Welcome everyone. I am Sugasini. I am assistant professor and working in University of Illinois at Chicago. Thank you for joining us today for Lipids and Health, exploring the roles of lysophospholipids and omega-3 fatty acids. This is a pre-event session of the 2023 AOCS Annual Meeting and Expo and brought to you by the Health and Nutrition Division of AOCS. We are streaming today's event onto the AOCS social media. So welcome to everyone watching there and here in Zoom. We have staff monitoring all streams. So please enter any questions you have for our speakers and they will be addressed at the end of each talk. I would like to introduce the first speaker. Our first speaker today is Dr. David Lawrence Silver. He is a professor and deputy director in the Signature Research Program in Cardiovascular and Metabolic Disorders at the Duke NUS Medical School, Singapore. Dr. Silver has a PhD in genetics and hold faculty position at Columbia University and the Albert Einstein College of Medicine, and his specialization in biochemistry of lipid metabolism. His long-standing research interests are in the areas of lipid storage, transport, and metabolism, and has a translational focus in applying his laboratory discoveries to the treatment of human diseases. And moreover, the Dr. Silver's laboratory is multidisciplinary, utilizing biochemistry, structural biology, molecular genetics in mice and humans, and molecular and cellular biology. His notable discoveries include the FATM family of endoplasmic reticulum proteins and MFST2A, a lysolipid transporter critical for human brain growth, and SPNS1. The SPNS1 is also a lysosomal lysolipid transporter, which is important for phospholipid salvage and lysosomal function. His laboratory research has been published in scientific research journals such as Nature Genetics, and PNAS. And I'll now turn over to Dr. Silver.
Thank you very much for that kind introduction. I'd also like to thank the AOCS for this opportunity to speak at your webinar. So as you just heard, I'll be talking about lysolipid transport. What you're seeing spinning in front of you on this screen is MFST2A, which is a sodium-dependent lysolipid transporter. And I'll tell you a little bit more about this. And I'd like to talk about its role in brain development and beyond, and I'll get to the beyond hopefully soon. What is MFST2A? It stands for Major Facilitator Superfamily Domain Containing 2A. It's a sodium-dependent LPC, or lysophosphatidylcholine transporter. It has high specificity for polyunsaturated fatty acids that are linked to LPCs, as well as monounsaturated fatty acids. And it's the primary route by which the brain acquires omega-3 fatty acids. And I think this provides the mechanistic basis for Michel Lagarde's and co-workers' findings of about 30 years ago, where they showed the preferential uptake by the brain of LPC conjugated to DHA, preferentially over unsaccharified fatty acids. So in the middle of the screen of the slide here, you're looking at an LPC molecule containing DHA, which is this fatty, greasy part here off to the left of my screen. And you have a zwitterionic head group. So you have choline, phosphate, and glycerol. When these are transported into cells, they're acted upon by a number of enzymes found in the human genome and other mammals called lysophosphatidylcholinesyl transferases, which utilize a fatty acyl-CoA, esterifying those to this free hydroxyl group. And essentially, you end up with a phospholipid, which is important, of course, for constructing membranes. And just keep that in mind for a moment. So I think we can consider that these LPCs that circulate on albumin in blood are membrane building block precursors. I'll get back to that in a moment. We recently published this paper with a collaborator of mine, Filippo Mancia at Columbia University, in which we solved a 3-angstrom cryo-EM structure of MFST2A. And we were fortunate to capture this structure with an omega-3 fatty acid containing LPC. You can see right here in the space-filling part of this structure. And I just want to make a couple of points here. So this line, of course, indicates the membrane in which this is sitting. This is outside the cell. This would be inside the cell down here. And we captured the LPC in an inverted position. And what I mean by that is you can see the head group here peeking out from the transporter, and this is the long acyl chain of the omega-3 fatty acid, in which case, this case is an 18-3 fatty acid. Two points I'd like to make. One is there's the transport cleft is extremely large for this protein. So it has a very large accommodation apparently for modifications to this acyl tail, and I'll tell you a little bit more about that in a moment. The other insight that this structure gives us, in addition to identifying how sodium or where sodium is binding and some other aspects of how the lipid is being bound within the cleft, it tells us that since this lipid is inverted and it looks like it's on its way out of the transporter, that this transporter likely works as an LPC flipase. And so you can see this in this very simplistic diagram of what we posit how this would be working. So you have the LPC bound to the outer leaflet of the membrane, somehow transiting through the transporter, flipping and inverting, and then transiting laterally out of the transporter. We recently developed an in vitro assay that tested this hypothesis and proved it to be true, that MFSC2A utilizes what we call a flipase mechanism. If those in the audience are interested in the details of how we did this using a proteoliposome purified system, I can refer you to our recent paper here at the bottom, which I will not go any further into. Together with clinical collaborators around the world, we've identified a number of unique mutations in the human MFSC2A gene, which is located on chromosome 1. All of the patients that have been characterized, we're talking about more than 13 families, exhibit very similar clinical presentations, and that is shown here: microcephaly and hypomyelination. So you can see a couple of the example pedigrees shown here at the bottom, including the T2 scans of brains from these patients. And all of them present with underdeveloped brains or microcephaly, as well as severe hypomyelination. I think we can explain the microcephaly based on that first principle that I showed you in the first slide, that the LPC serves as a building block for membranes. And so we know from preclinical studies that DHA deficiency itself does not result in microcephaly. So it's very likely it's the full complement of plasma-derived LPCs that are important for generating membranes in the brain. And so we have a number of papers that support that hypothesis, and I won't go into that further. Regarding hypomyelination, those are ongoing studies, so I won't be talking about that today, but suffice it to say that MFSC2A is expressed in oligodendrocyte precursor cells, which are important for myelination. And so we'll have a story out quite soon on that I'll be able to share. So to the beyond. The question that we had was, are there other physiological processes that require LPC transport, or is MFSC2A the only one? And so are there other such transporters that are similar to MFSC2A that can transport LPCs across membranes? Where would we look for this kind of process? We decided to examine what the lysosome. And why the lysosome? Well, because the lysosome receives a large amount of phospholipid from two major processes. One is endocytosis or phagocytosis, and the other is autophagy. So we imagine that the lysosome would have to dispose of phospholipids to maintain lysosomal homeostasis, lysosomal size, lysosomal function. Where do those lipids go? So we hypothesized that likely there would be another MFS a transporter that is specifically located in the lysosome to handle incoming phospholipid. So looking at an old paper from Christian de Duve in 1969, in Christian de Duve's lab at Rockefeller discovered the lysosome, and he was interested in understanding the biochemical fates of phospholipids within the lysosome. And so just to summarize some of his findings with this diagram here, he showed back in 1969, that phosphatidylserine, phosphatidylglycerol, phosphatidylethanolamine, and phosphatidylcholine, all shown here in abbreviations, are acted upon by phospholipases, whether they're A2s or A1s. There's a mixture of these in the lysosome, but they end up with a pool of lysolipids derived from these phospholipids. Ultimately, lysophosphatidylserine, lysophosphatidylglycerol, as well as lysophosphatidylinositol, which is not shown here, get further degraded or hydrolyzed by phospholipase to produce glycerol phosphoserine, glycerol phosphoglycerol, and as well as this pool of LP and LPC, to a smaller extent, get hydrolyzed to GPE and GPC, where just recently in a paper by Lactome in Nature implicates the putative transporter CLN3 in the removal of these glycerol phosphoserine or glycerol phosphorylate esters back to the cytoplasm. So we hypothesized that since Christian de Duve showed that it's the LP and LPC that accumulate in the lysosome and aren't efficiently broken down into these glycerol phosphorylate esters, that there should be a transporter at the lysosomal membrane that would mediate efflux of LPCs that would then enter into the pool, the cytoplasmic pool of phosphatidylcholines, again, through the action of these endoplasmic reticulum LP paths. So what is the identity of the lysosomal LPC transporter? And so based on essentially comparative biology, comparative biochemistry in evolution that MFSE2A mediates LPC transport, we hypothesize that the transporter probably belongs to this family of MFS proteins. And that's this paper that was recently published by my laboratory. And I'd like to go into some of the details here because there's some interesting comparisons to MFSE2A that I'd like to make. So how did we discover that it was SPINS1? Well, we used a screen that takes advantage of the normal cell biology by which transporters, membrane transporters, reach the lysosome through the secretory pathway. And they do that through synthesis at the ER, transport to the Golgi. Let me just change my laser pointer. I think it's going to be easier to see. Through the Golgi, ultimately reaching then the plasma membrane, they undergo endocytosis and they reside in the lysosome. So we took advantage of this normal route of transport of membrane proteins on their way to the lysosome to measure their function once they reach the plasma membrane. You can do that by lowering extracellular pH. You can add radioactive LPC and then measure the activity of the MFS transporters. And so this is the list of MFS transporters that we looked at. All of them are considered orphans. That is, we don't know what ligands they transport. And lo and behold, in our assay using C14 LPC oleate, SPINS1 was the only one in this group of known lysosomal transporters to transport an LPC. Okay. We tested sphingosine. Given the nomenclature of SPINS, it was considered a putative sphingosine transporter. That's not the case. It does not transport sphingosine, and I'll show you with more analysis that it doesn't transport lipids containing sphingosine as a base. We did a structural model of SPINS1, and we found some interesting, I would say some unique features that are different than MFSE2A. One, it's not a sodium-dependent transporter. Second, the large pocket or transport cleft for LPCs that is seen in MFSE2A is not seen in SPINS1. SPINS1 is a very tight cleft, as you can see here, where this yellow line signifies the acyl tail of the LPC. So it's a very tight fit in the hydrophobic pocket. So the pocket's very small. And we identified putative head group binding residues an arginine 76, a glutamate 164, and a histidine 427. And just to highlight that this is a very tight fit, in our experiments, we're not able to see very robust transport of any LPC conjugated to fluorophores. That is, if you conjugate a fluorophore, such as NBD, to the omega carbon of this acyl tail, SPINS1 really does not transport it. So it has extremely low affinity for any kind of LPC conjugates, consistent with the very tight fit that this pocket forms around the acyl tail. So we've made mutants in these potential head group coordinating residues, and most of these are dead. They do not transport. And what this graphs show you is that SPINS1 wild type can transport LPC oleate, LPC DHA, LPC palmitate. Doesn't seem to have a lot of specificity for the acyl tail, in fact, whereas MFSE2A has a lot of specificity for the type of acyl tail. Since this is a lysosomal protein, we did a pH response, activity response, and SPINS1 is exclusively functional at lysosomal or near lysosomal pHs, consistent with a proton-driven process. The bottom graph shows a dose response typical for an MFS protein or a transporter, a facilitative transporter. Now, we then developed a mass spec-based approach to study the types of lipids that SPINS1 can transport. And so this is a nice approach because we're not modifying the lipid, we're using native lipids. So we call this a structure-activity relationship analysis. So we incubate the various lipids shown here in this heat map. We call this lysolipid for treatment. These are the ones that were added to cells in culture. You can see that on the y-axis here. Then within an hour, we extract the lipids and measure both the lipid that was added as well as the first product in metabolism. So we call this the bioincorporated lipids. That is, so if you look at LPC 22,6, that gets essentially esterified with either a 16-carbon fatty acid or an 18,1 carbon to produce these two phosphatidylcholines, 38,6. And again, if you're not familiar with the nomenclature, the first number is the number of carbons. The second after the colon is the number of double bonds. Okay, what did we find? Comparing to MFSE2A and a control, which is our histidine 427 alanine mutant, which is dead for LPC transport, we see that there's a lot of similarity between what MFSE2A and SPINS1 can transport with several exceptions. One is MFSE2A can transport LPI and LPS, whereas SPINS1 does not. And you can see down here at the bottom, none of these neither of these two transporters transport lysophosphidic acid, nor do they transport lyso-SM. That's also called sphingosine phosphorcholine. So again, despite having the same head group, the phosphorcholine head group, but in this case having a sphingosine backbone, SPINS1 does not transport this. Okay, and this is not a lipid that just freely passes through the plasma membrane. And then you can see that these are all bioincorporated. So just to summarize, MFSE2A additionally transports LPS and LPI. So it does have some differences in what it can recognize in terms of the head group. And again, MFSE2A, but not SPINS1, can transport LPCs conjugated to fluorophores at the acyl tail. I didn't show you this data, but this is in most of our biochemistry papers where we study MFSE2A transport. We always use these kind of fluorophores to study its activity. What's known historically about SPINS1? We're not the first to discover SPINS1 by far. This has been around for at least 2 decades. It's evolutionarily a conserved protein found in lysosomes from worm to human. It's ubiquitously expressed in all tissues and cells in mammals. These are the model organisms that have been studied in the past. There's some very common features of phenotypes that are seen in loss of function. That is negative impact on survival. That's also true in mice. They all knockdowns or knockouts in these organisms have enlarged lysosomes, increased lysosomal pH, and defective autolysosomal formation and lysosome reformation. So it's always been known that SPINS1 is a lysosomal protein and it's critically involved in lysosomal homeostasis. When we knocked out using CRISPR-Cas9 SPINS1 in HEC293 cells, we can recapitulate most of these findings from the literature. That is, you see very large enlarged lysosomes as denoted by lysocracker in the knockout lines versus control. Even under basal conditions in this Western blot analysis, we see an increase in LC3B2, which indicates to us that there's a defect in autophagy. So you can see just under normal conditions compared to normal conditions here in the control, LC3B2 is up and it remains very high when we activate autophagy. So that's consistent with previous findings that SPINS1 deficiency results in defects in lysosomal dynamics. We then went on to incubate cells with C14 choline, the structure shown here. So we can incubate these overnight to label up steady state the phosphatidylcholine pools in a cell. We can purify lysosomes using superparamagnetic iron beads that were fed to the cells. And you can see in alternating lanes on this thin layer chromatography image, so you have control and knockout, that there's a very large increase in total LPCs in cells lacking SPINS1. So this is the first proof that SPINS1 in cells is required for removal of LPCs from the lysosome. We did high resolution mass spectrometry to see what kind of LPCs in other species of lipids accumulate, and lo and behold, the vast majority of lipids that are accumulating. And on the y-axis, this is you can see the abundance of these lipids in the cell. And the vast majority of lipids that accumulate, the significant ones that are shown as colored dots, the gray ones are not significant, are LPCs. You can see that the lower abundant accumulation of other lipids, namely sphingosine, two species of sphingosine shown here, but then again, sphingosine nor sphingomyelin is transported. So this is a secondary effect. We can quantify each of these LPCs. You can see total LPC is accumulated when we re-express a wild type SPINS1. So this is a rescue experiment. We can reduce lysosomal total LPC. And this is true for all the different LPC species that we measured. So the largest changes in lipids found in a SPINS1 knockout cell, mammalian cell, are the LPCs. But to prove in vivo in an intact cell that SPINS1 mediates efflux required another type of experiment, which is a stable isotope flux experiment that we designed. And we did this by using deuterated, this is D9, so the methyl groups are now deuterated at the head group of a PC, and this PC has two oleate tails, acyl tails, and we can learn from the gene therapy field how to make nanodiscs and deliver those to the lysosome. And so we used an ApoE coated nanodisc containing D9PC36-2, and this allows interaction with the LDL receptor and endocytosis into the lysosome. And you can see in our paper, we can show lysosomal enrichment versus using discs without ApoE. Nonetheless, if a cell, the hypothesis is if a cell expresses SPINS1, then through the action of PLA2 in the lysosome, LPC18-1 would be formed. This would be effluxed out of the lysosome via SPINS1, and then reesterified by various common fatty acids or abundant fatty acids in these cells. mixing up that acyl tail or acyl tails to PC32-1, 34-1, and 34-2. So this would be evidence that SPINS1 is mediating efflux out of the lysosome based on this analysis. In the absence of SPINS1, we would predict a very large accumulation of D9-LPC and a reduction in the production of these downstream reesterified LPCs. And so when we did this experiment over time, because this is a flux study, 1 hour, 3 hours, and 6 hours, you can see that total labeling of cells, regardless of having SPINS1, is the same compared to control. So there's no defect in LDL or LDL receptor pathway for the uptake of these nanodiscs. However, over time, you can see that the knockout cells have a robust accumulation of LPCs, consistent with the transport data that I showed you. Now, if we look then at, and I should say the control cells don't do that at all. They do not accumulate at all. Again, consistent with the existence of SPINS1 in the lysosome mediating efflux. So then looking at the downstream products, you can see that the knockout cells for any of these identified PCs with different acyl tails now, but still have a D9 choline head group, they're reduced for the knockout. It's not a complete inhibition of conversion to these downstream lipids, but it's a very significant drop among all of them. And the reason which we show in our publication, we describe why this is the case, why this doesn't look like this graph here, where the knockout still has bioconversion, is because the nanodisc doesn't 100% go to the lysosome. Some of the nanodisc fuses with the plasma membrane. So this is a kind of a background that you're seeing. Nonetheless, you can see that the wild type has a much higher conversion rate of LPC 18-1 into these various PCs. So what about multicellular organisms? Is this true in multicellular organisms that SPINS1 mediates efflux of LPC? So we went back to the model organism zebrafish, which was well described in the literature by Kishi at Scripps many years ago. And the salient feature of SPINS1 knockdowns, or we call these morphins, is that the yolk sac becomes non-transparent, so it's very opaque. And when we think about yolk sacs, we think about lipids. The yolk sac itself is a bag full of lysosomes, which contain lipids. And these lipids during embryonic development of the fish, get transported to the body of the fish. So this phenotype in hindsight makes a lot of sense to us. And so when we did the same kind of high resolution lipidomic analysis, and again, this is a lipidomic abundance plot, you can see that the only significant elevated lipids in the fish, in this embryo, are the LPCs. In this case, we didn't see any increase in sphingosines at all, or other lipids. Again, suggesting those are all secondary effects. And then we can quantify this. So among all the different LPCs, whether we're looking 24 hours after fertilization or 56 hours after fertilization, the SPINS1 morphins have accumulation of LPCs, consistent with the cell-based studies. What about liver? So let's go up the evolutionary chain now to mammals. So we use an AAV shRNA approach to knock down SPINS1 in the liver. We chose the liver because the liver has very active lysosomes handling lipid all the time, since that's really the central hub in the body where LDL is disposed of. So one of the first phenotypes that we saw was that there's likely liver damage. So some of the liver enzymes like alanine aminotransferase are significantly elevated in animals that have deficiency in SPINS1 in the liver. There's a slight but significant increase in circulating cholesterol, no change in triglyceride. And then looking histologically at liver sections from knockdowns versus controls, looks very much like what we see in cell culture for knocking out SPINS1. We have an enlargement of lysosomes denoted by LAMP1 positive staining or cathepsin B positive staining, and quantification shown here at the right. So so again, knocking out or knocking down SPINS1 in cells, and as well as liver, results in the same phenotype, enlargement of lysosomes, as well as defects in autophagy. So again, you're looking again at a marker for autophagy, LC3B2, and in each lane is one animal. You can see that there's an accumulation of LC3B2, so indicative of defects in autophagy. Running an RNA sequencing analysis to get more information about what's going on in the liver, we can see that the top pathways that are elevated in the livers of knockdowns are those involved in inflammatory pathways. So knocking down SPINS1 in the liver results in a very clear liver pathology. It's not a fatty liver disease. It's looks more like an inflammatory disease. So then running high resolution mass spectrometry on both isolated lysosomes from knockdowns versus controls from these animals versus total lysates, you can see across the board that many of these lipids are significantly elevated in lysosomes. And in some cases, this can even be seen in total liver lysate. And that includes ceramides, hexosylceramides, sphingosines, sphingomyelin, gangliosides, and this very important and specific marker for the lysosome, BMP. This is a very unique lipid for the lysosome. You can see it's significantly elevated. This indicates often that the lysosomal compartment is expanded. And so this looks, I should just also mention here that cholesterol ester is also, cholesterol ester is also increased, in addition to very large increases as expected in LPC, LPE, LPS, including LPG. So this looks very much like a lysosomal lipid storage disease, similar to Niemann-Pick C type 1. So when we look then at the individual species comprising the LPC and LPE lipids, you can see these are the most elevated out of all lipids when we knock down SPINS1. So all of them are elevated, and in some cases you can even see these elevations just in total liver lysate, but they're highly enriched in lysosomes. Okay, so this is very consistent with the previous data that I showed you, that SPINS1 mediates efflux of LPCs. So to summarize, back to this original slide on the lysosome, the breakdown products of PS, PG, PE, and PC result in accumulation of these pools of the lysolipids, in some cases, particularly LPS, LPG, and a little bit on this side for the LP and LPC, result in complete hydrolysis down to their head groups, or the glycerol phosphodiesters. And this leads to efflux through an unknown mechanism involving CLN3. And in the cytoplasm, there's phosphodiesterases that release glycerol 3-phosphate for recycling, as well as choline coming from this side here. And that can be recycled back into cytoplasmic phosphatidylcholine pools through de novo phospholipid synthesis, and that's the Kennedy pathway. SPINS1, on the other hand, mediates the efflux specifically of LPE and LPC from the lysosome, contributing to phospholipid pools. And so we can conclude that SPINS1 contributes to membrane phospholipid homeostasis through LPC and LPE recycling and from the lysosome, and is critical for cellular homeostasis. And again, in animals, the major phenotype that we've seen is liver inflammation. So I'd like to end here and thank the members of my lab and my collaborators, particularly Menglen He and Alvin Cook for all the SPINS1 work that I showed you. I'd also like to thank Federico Torta and his laboratory for performing the high resolution lipidomic analysis for us, as well as many of my collaborators and clinical collaborators throughout the year. And thank you for your attention, and I'm looking forward to answering your questions.
Thank you, Dr. Silver, for the very informative presentation. not only MFS-DTA, SPNS1, lysolipid transporter, which plays an important role for phospholipid salvage and lysosomal function. Open for the questions.
Sugasini, should I read this or you would like to read it?
Yeah, you can read it.
Okay, so there's a question. Is it likely that DHA-LPA is produced in plasma by PLA1 lipases? In addition, most DHA in phospholipid molecules are attached to the SN2 position of the glycerol backbone. My question is, does the transporter distinguish SN1 and SN2? I see. Okay, so is it? So I think so it's not LPA. Maybe the questioner is LPC. Is it likely that DHA-LPC is produced in plasma? There's some evidence actually from Sugasini's lab that some of these lipases, such as hepatic lipase and I think it's the endothelial lipase, is involved in production of LPC DHA in the plasma compartment. So that probably could constitute part of the production of LPC DHA. The second question is whether SN1 or SN2 is important. It doesn't seem to be at all important for transport by MFSC2A, and there's no structural reason that we could see from our structure, nor the other two structures that have been published on MFSC2A. Thanks for your question.
Any questions? Thank you, Dr. Silver. We are moving on to the next speaker.
Thank you very much.
Our next speaker is Dr. Undurti N. He is the CEO of UND Life Sciences and an adjunct professor. He received his Bachelor of Medicine and Bachelor of Science from Andhra Medical College in Visakhapatnam, India, and his medical degree from Osmania Medical College in Hyderabad, India. He is known for his research in the field of rheumatology and immunology. He has several patents in the United States and India, and he is the author of multiple books. He is a fellow of the National Academy of Medical Sciences in New Delhi and also a fellow of the Royal Society of Chemistry in London. Please welcome Dr. Das.
Thank you very much. Are you able to see my slides?
Yeah.
Okay. Okay, that I just, and able to hear me?
Able to hear nicely.
Okay, thank you very much. So good afternoon, everybody. After an excellent lecture by the previous speaker on omega-3 fatty acids and their transport. Now I'm going to talk to you about a less appreciated and less well-articulated topic like arachidonic acid. Because most of the research work, especially in N3 and 6 fatty acids, is all devoted to N3 fatty acids. Whereas I'm going to talk to you about arachidonic acid, which is an N6 fatty acid. This work started back in 1980s when we found that when we tested all the fatty acids in the patient's plasma who have diabetes, we found that it is significantly lower. Arachidonic acid is significantly lower. And so we thought that we should concentrate on arachidonic acid, unlike which others are looking at N3 fatty acids. As many of you may know, arachidonic acid is a 20 carbon and 4 double bond M6 fatty acid. It is present in our diet, but it is present in very small amounts. And so if you want to have adequate amounts of arachidonic acid in the body, it has to be converted from the dietary essential fatty acid, namely linoleic acid, which is 18-2. And so we think that most of the arachidonic acid that one measures is coming mainly from the endogenous formation. And this conversion of LA to AA occurs by the action of several enzymes. And most of the important enzymes are delta 6 and delta 5 desaturases. And then there are also elongases. So when you look at the biochemistry of linoleic acid conversion to arachidonic acid, linoleic acid is converted to gamma linoleic acid by delta 6 desaturase. Then it is elongated to form dihomo gamma linoleic acid, DGLA. which is 20 carbon, 3 double bonds. Then DGLA is converted to arachidonic acid by delta 5 desaturase into 24, which is arachidonic acid, and whose molecular weight is only 305. This is the pathway I am showing you here. And for comparison, you can see both N3 and N6 fatty acids, and both are acted upon by the same set of enzymes. And so people think that the both arachidonic acid metabolism and the N3 fatty acids, namely EPA DHA metabolism, are very closely linked. But from our research, we think that there are probably certain isoenzymes of delta 6 and delta 5, because there is no parallel increase or decrease in the EPA DHA and compared to arachidonic acid. So this when you look at the arachidonic acid metabolism, delta 6 desaturase is the rate limiting enzyme, and similarly delta 5 also is a rate limiting enzymes. So measuring these enzyme activities are extremely important to know what is happening to the dietary linoleic acid and its conversion into arachidonic acid. Back in 1993, that we started looking at these fatty acids in patients with several diseases like essential hypertension, then diabetes mellitus, and coronary artery disease. And when I say diabetes mellitus, it is type 2 diabetes, which is not insulin dependent. So we measured at that time the uh, plasma phospholipid fraction, the fatty acid distribution in these patients. And then we found very interesting changes. And this is a slide showing the levels of various fatty acids in the plasma phospholipid fraction of these patients. And you find that in the fourth column, that in type 2 diabetes mellitus, there is not much, there is not much change in the linoleic acid, but there is a decrease in arachidonic acid, almost 50% decrease in the arachidonic acid. So showing that there is a very substantial decrease in arachidonic acid, very specific decrease in arachidonic acid. There is also decrease in the DHA from 1.4% to 2.5%. So there is a very drastic decrease in the levels of arachidonic acid and DHA in patients with diabetes mellitus. Of course, in other types of patients also, there are changes in arachidonic acid. In fact, arachidonic acid levels are decreased in all diseases, right from hypertension to coronary artery disease to diabetes, type 2 diabetes, and diabetic nephropathy. So suggesting that all these diseases are probably have an abnormality in arachidonic acid metabolism. And this is important because most of these diseases occur together in patients. So the patients who are obese, who have hypertension, also end up developing type 2 diabetes. And patients who have type 2 diabetes, they end up developing hypertension, either prior to the development of type 2 diabetes or after some time after developing the type 2 diabetes. And all these patients, both the type 2 diabetes and hypertension patients, have very high incidence of coronary artery disease. So suggesting that there is a common denominator in the form of decreased arachidonic acid in these patients. And this is very important. So this suggested to us that we should concentrate on arachidonic acid and see what is happening in the development of diabetes in animal models and also in patients. So this is an in vitro study where we took rim cells, that is rat insulinoma cells, which are known to secrete insulin. And so we took those cells and treated them with various concentrations of alloxon, which is known to be toxic to pancreatic beta cells. And you find that as you increase the concentration of alloxon, there is a decrease in viability of the pancreatic beta cells. And of course, we did also a time course study. And so once we have established the dose of arachidonic alloxon that can decrease the survival of these pancreatic beta cells, then we tested several fatty acids, but I'm showing you here the slide with arachidonic acid because we found that arachidonic acid is the most effective one. So arachidonic acid has increased by itself does not have any effect on the survival of the pancreatic beta cells. So it is not toxic even up to 48 hours of incubation with 15 micrograms of arachidonic acid, pure arachidonic acid. But when you take alloxon and then the cell viability is decreased and you add arachidonic acid, the cell viability is increased. So, suggesting that as increase the concentration of arachidonic acid and the presence of alloxan, the viability of the rin cells is increased, or it has come to almost normal levels, suggesting that arachidonic acid has a cytoprotective action on pancreatic beta cells. So this coupled with the information that arachidonic acid levels are low in patients with type 2 diabetes suggests that the lower arachidonic acid levels may probably be responsible for the decreased survival or decreased number of pancreatic beta cells in these patients. So arachidonic acid is a important cytoprotective molecule for pancreatic beta cells. Then we repeated this study using streptozotocin, which is also another pancreatic beta cell toxic chemical. And then we found that streptozotocin also decreases the viability of the rin cells. And when you add the arachidonic acid different concentrations, it is able to increase their survival. So suggesting that arachidonic acid is cytoprotective both against alloxan and streptozotocin. And then, of course, we did two different types of experiments in this model, where we pre-treated the cells with arachidonic acid and then added streptozotocin or alloxan. And then we added simultaneously arachidonic acid and alloxan or streptozotocin. And we found that in both instances, the arachidonic acid is able to protect the pancreatic beta cells against the cytotoxic action of alloxan and streptozotocin. I'm not showing those slides, but we also looked at several other chemicals like 4-alpha farbol, then diphenylhydrantoin, and certain anti-cancer drugs like doxorubicin. And there also we found that they all decrease the survival of pancreatic beta cells, and they are protected by the arachidonic acid by simultaneous or pre-treatment with arachidonic acid to the pancreatic beta cells, suggesting that arachidonic acid seems to have a cytoprotective action. In general, people in the lipid area think that arachidonic acid is a toxic chemical, and so many people uh, don't do many experiments with arachidonic acid. But paradoxically, our results showed that arachidonic acid is not toxic to normal cells, and in fact, it is protective to the pancreatic beta cells. Then, of course, as I already told you, we wanted to find out whether arachidonic acid by itself is protective or it has to be converted into prostaglandins or leukotrienes or some of their metabolites. So this is a slide showing where we looked at the by blocking the cyclooxygenase enzyme and lipoxygenase enzymes using various chemicals, and we found that whether you block these cyclooxygenase or lipoxygenase enzymes or not, still arachidonic acid is able to protect the pancreatic beta cells against alloxan and also streptozotocin, suggesting that the fatty acid itself is a cytoprotective. It does not have to be get converted into prostaglandins or leukotrienes. This came as a surprise to us because most of the time we think that arachidonic acid has to get converted into its metabolites like prostaglandins, leukotrienes, and so on and so forth. And those metabolites are they're ones responsible for the various actions that are observed with arachidonic acid. But we found that even if you block both cyclooxygenase and lipoxygenase enzymes, still arachidonic acid is able to protect the pancreatic beta cells against alloxan and streptozotocin induces cytotoxicity, suggesting that the arachidonic acid itself is cytoprotective, or it is possible there is some other product that is formed. Sorry, there is some other product that is sorry, what happened? Some other product that is formed from arachidonic acid, which is able to protect the pancreatic beta cells. So we will come to that a little bit later. Then, of course, just to confirm this, we also performed studies with various prostaglandins and leukotrienes. And I'm not showing those slides, but we showed that various prostaglandins and leukotrienes do not protect the pancreatic beta cells against alloxan and streptozotocin, suggesting that arachidonic acid itself is able to protect the pancreatic beta cells. Then with this in vitro results information, we thought that we should extend it to the in vivo animal model and see what happens. So this is a slide showing where we induce type 1 diabetes using alloxan. This slide, we had done experiments both with alloxan and streptozotocin, but this is a slide showing with streptozotocin. And we injected streptozotocin and induced type 1 diabetes. And you see that in streptozotocin, there is a very large increase in the plasma glucose levels up to 300 to 400. Whereas when we treated them with arachidonic acid, either intraperitoneal or oral. See, this is surprising because most of the time the experiments with fatty acids are done, pure fatty acids are done either intraperitoneal injection because people want to avoid any absorption issues. So in this study, we gave arachidonic acid intraperitoneal or oral and the same amount of dose. Both intraperitoneal dose and oral dose was the same, and we found that the effect is same. So whether you give arachidonic acid orally or intraperitoneally, it is able to decrease the severity of the diabetes. And we studied this experiment for 30 days till the end of the day. And then, of course, we looked at the body weight, and the body weight comes back to near normal levels in the arachidonic acid treated animals. So this suggests that arachidonic acid, whether given orally or intraperitoneally, is able to prevent the development of diabetes induced by streptozotocin and alloxan. Then, of course, we wanted to see confirm with this by measuring the plasma insulin levels. We find that the plasma insulin levels are high compared to the streptozotocin alone treated animals, and plasma levels, insulin levels are almost similar, both in the oral group and the intraperitoneal group. Then, because we believe that nowadays the diabetes is a inflammatory condition, so we looked at the plasma TNF alpha levels. They are very high in streptozotocin treated animals with where type 1 diabetes is induced, and we find that these TNF alpha levels drop. They don't come back to normal levels, but they are substantially reduced by treatment with arachidonic acid, suggesting that arachidonic acid has anti-inflammatory action. Then, of course, we wanted to extend it this and see whether the same results can be obtained with type 2 diabetes. So we used again, this is the protocol which we followed. We used streptozotocin to induce type 2 diabetes in the Wistar rats. And then we gave arachidonic acid per 7 days, continuously every day. And then once in a week, oral arachidonic acid was given. So the idea is that initially we wanted to top the body with a sufficient amount of arachidonic acid and the weekly supplementation of arachidonic acid and see what happens. Then, of course, we did all the experiments, look at the plasma glucose levels, insulin, and so on and so forth. We taken the several tissues and then we look at all those tissues. Here you find that the plasma glucose levels are extremely high in patient in the type 2 diabetes animals. And then, of course, we measured it on 3 occasions, on 10th day, 20th day, and 30th day of the experiments. And then, of course, 10th day also it is high, but by 20th and 30th day, the levels go up to almost 400 milligram percent. Arachidonic acid by itself does not have any effect on the plasma glucose levels, but arachidonic acid with streptozotocin treatment, they produce a substantial decrease in the plasma glucose levels, suggesting that arachidonic acid can prevent even type 2 diabetes. Then, of course, we looked at the body weight. Body weight is attained to normal in the arachidonic acid treated animals. Then we looked at because one of the problems in patients with the type 2 diabetes is they have insulin resistance. That means their pancreas may be producing sufficient amount of insulin, but that insulin is not able to produce its action of driving the glucose into plasma glucose into the cells. So that is because of insulin resistance, and that insulin resistance is supposed to be because of the pro-inflammatory molecules like IL-6 and TNF-alpha increase. So in these animals, we measured the plasma insulin resistance indices, and we found that in treated type 2 diabetes animals, that the insulin resistance is increased, whereas when these animals are treated with arachidonic acid, the insulin resistance is decreased very substantially, suggesting that insulin resistance can be reduced by arachidonic acid treatment, and this may partly be because it is able to reduce the TNF alpha and IL-6 levels in these animals. Then, of course, in these animals, again, we measured the plasma insulin levels. The insulin levels are increased compared to the control, and they are decreased compared to the STZ-induced type 2 diabetes animals. This is because they don't need, these animals do not, which are treated with arachidonic acid, do not need more insulin because insulin resistance is decreased. So with less insulin, you get the same amount of action. So suggesting that arachidonic acid is able to reduce insulin resistance, and so the pancreas does not have to produce more insulin levels, and still the plasma glucose levels will be near normal levels. This is a slide showing the, as I was telling you, plasma interleukin 6 levels and TNF alpha levels, and you find that both levels are decreased compared to the STZ treated animals, suggesting that the arachidonic acid treatment can reduce the inflammatory molecular levels and so reduce the insulin resistance. Then, of course, we measured what is called as oral glucose tolerance test, because whenever a person who is obese and who has a family history of diabetes comes to the physician for assessment of his uh, plasma glucose levels or whether he is prone to develop diabetes, we do a test what is called as oral glucose tolerance test. That means the patient is given 75 grams of glucose orally, and then at various periods of time, we measure the plasma glucose levels and see what is happening. So the plasma glucose levels should not cross 200 milligram per set any time. And in a normal person, the plasma glucose levels do increase from the fasting level, but they would eventually be less than 200 milligrams. And this is done for about generally for 2.5 hours to 3 hours. In this instance, we have done up to 2 hours. And then we measured the plasma glucose levels and insulin levels. So there will be a corresponding change in the plasma glucose levels and insulin levels. As the plasma glucose are increasing, the pancreas tries to produce more insulin to control the glucose levels. So in this experiment, you find that the animals that are treated with arachidonic acid have almost normal oral glucose tolerance test, suggesting that there is no insulin resistance or a decrease in insulin resistance. And the insulin production need not be increased by the pancreas because plasma glucose levels are low. And this is partly because of the insulin resistance that is decreased by arachidonic acid treatment. And that is because the inflammatory molecules IL-6 and TNF are decreased by the arachidonic acid treatment. Then, of course, we wanted to see whether there is what happens to the LOX enzymes and COX2 enzymes, then NF-kappa B expression in these animals, because we saw that IL-6 and TNF-alpha levels are decreased, and so we wanted to look at the NF-kappa B expression and see what happens. And you find in the last column that the NF-kappa B expression is a decrease in animals which are treated with arachidonic acid and streptozotocin compared to only streptozotocin treated animals. That means streptozotocin is able to induce significant amount of inflammation in the form of increased expression of NF-kappa B, and that is decreased by the arachidonic acid treatment, and it is almost equivalent to the control levels. Okay. Whereas the more important observation here is when only arachidonic acid is given, the control animals which received arachidonic acid, they also showed even a further decrease in NF-kappa B expression compared to the control. So suggesting that even under normal conditions, if there is enough amounts of arachidonic acid, it can suppress the NF-kappa B expression and so decrease the inflammation process. So it looks like one of the normal functions of arachidonic acid in the body would be to control or to regulate the inflammatory process by decreasing NF-kappa B expression. Of course, then we looked at the 12-LOX, 5-LOX, and then COX2 enzymes. And in our experiment, we did not find any 15-LOX expression. We find that there is some amount of decrease in 12-LOX expression and in 5-LOX expression, but it is not very substantially different. That means the LOX expression is not very much different from the treated STZ animals, but there is some amount of decrease, but it's not very, very significant. Most important is the 5-LOX expression is equivalent to almost the control levels, whereas the 12-LOX expression is still higher compared to the control. When we look at the COX2 expression, there is no change. So suggesting that when we give arachidonic acid, there is a very substantial decrease in NF-kappa B expression, decrease in IL-6 and TNF-alpha levels, with little changes in COX2 expression. In fact, there is a slight increase, but it is not actually significant, and very slight change in the LOX expression and in the 12-LOX expression. That means these enzymes are not substantially decreased, suggesting that if the cells want to utilize the arachidonic acid that is fed to the animals to form leukotrienes and prostaglandins, they can do so. So that is very important. Whereas in our in vitro experiments, we found that when you block COX2 and LOX enzymes, then still arachidonic acid is able to produce its its cytoprotective action against alloxan and streptozotocin. So suggesting that even though the enzymes of 15 logs, 12 logs, and COX2 are not decreased by the arachidonic acid treatment, there's only a decrease in the inflammatory gene expression, like in the form of NF-kappa B decreased and decreased IL-6 and TNF-alpha. So there is no substantial inhibition on prostaglandin and leukotriene synthesis in the presence of arachidonic acid. This came as a surprise to us. And then, of course, we took the adipose tissue and then we looked at the adipose tissue also. We found that in even adipose tissue, there is decreased NF-kappa B expression. 12-lox expression is comes back to normalcy. And this is important because in diabetes, they have more adipose tissue and adipose tissue inflammation is not present, suggesting that whatever arachidonic acid we have fed to the animals is able to decrease adipose tissue inflammation, the LOX expression. And then lipocalin production expression also is decreased, suggesting that adipose tissue inflammation is substantially decreased by the arachidonic acid that is fed to these animals. Then we thought that if there is no role for prostaglandins and leukotrienes, is it possible that substantial amounts of lipoxin F4 are being generated by the arachidonic acid that is fed, which is responsible for the anti-inflammatory action which we are seeing in these type 1 and type 2 animals. So what we did is we measured the plasma lipoxin F4 levels, and we found that lipoxin F4 levels are substantially increased in these animals. So that means whatever arachidonic acid we are giving, it is not giving rise to prostaglandins and leukotrienes, but it is able to get converted into lipoxin F4, which is responsible for the anti-inflammatory action of arachidonic acid. Because lipoxin F4 is a potent inhibitor of NF-kappa expression, it can decrease IL-6 and TNF-alpha levels, and it can also decrease adipose tissue inflammation. So whatever observations we made, both in vitro and in vivo, can be explained because of the increased generation of lipoxin F4 by the arachidonic acid that is fed. So it is possible that in these patients with type 2 diabetes where arachidonic acid levels are low, because the low levels of arachidonic acid, they may not be forming substantial amounts of lipoxin F4, and so they have inflammatory process in the form of low-grade systemic inflammation, and that is why they have increased levels of IL-6 and TNF-alpha, and that is responsible for the low-grade systemic inflammation that is seen in patients with type 2 diabetes. So with this evidence, because we now know that, of course, we have done the in vitro studies also, I am not showing those slides, we found that even in vitro, there is no increased formation of PGE2 and lycotrienes, but there is increased formation of lipoxin F4 from green cells when treated with streptozotocin, alloxan, and in the presence of arachidonic acid. Then we wanted to see whether this is true of in patients. So we took patients with the type 2 diabetes, and we also have, of course, incidentally, retinopathy, non-proliferative diabetic retinopathy, and proliferative diabetic retinopathy. So then we measured the lipoxin F4 levels in their plasma and also in their vitreal fluid. I think I have a slide about that. So in these patients, in the serum, we measured several other parameters, and then we measured lipoxin F4 level. And you find that as the disease is progressing, with just only diabetics without any retinopathy, non-proliferative diabetic retinopathy, and proliferative diabetic retinopathy, as the disease is progressing, there is a substantial decrease in the levels of lipoxin F4. That means as the diabetes disease and these complications of diabetes are increasing, then the plasma levels of lipoxin F4 is falling, showing that the increase in the inflammatory process is much more significant as they develop retinopathy in these patients with diabetes. And of course, we also measured other parameters. I'm not going to discuss about them, but this is important because that means the patients with the type 2 diabetes have low plasma arachidonic acid levels, and they have low levels of lipoxin F4. And so that may be responsible for the low-grade systemic inflammation seen in these patients. So in general, this is a summary slide. I will just summarize. So in general, people think that arachidonic acid is a pro-inflammatory molecule, and so many people do not do any investigations on arachidonic acid. They concentrate only on N3 fatty acid. But our in vitro results and in vivo studies suggested that arachidonic acid is not really a pro-inflammatory molecule, but in fact, it has anti-inflammatory molecule. It can prevent the cytotoxic action of several chemicals like alloxan, streptozotocin, doxorubicin, 4-alpha-farbol, and so on and so forth on several cell lines. And then that is not dependent on the production of prostaglandins and leukotrienes, but that is dependent on the ability of the cells to convert arachidonic acid into lipoxin A4. And arachidonic acid decrease the synthesis and release of TNF alpha and IL-6 and NF-κ expression in this in both in vitro and in vivo levels. And so we think that one of the mechanisms by which high-fat diet induces type 2 diabetes is because this is able to inhibit the conversion of linoleic acid into arachidonic acid by acting on delta desaturase. And because of low arachidonic acid levels, lipoxin A4 is not formed in substantial amounts, and so they develop the diabetes. Thank you very much. Any questions? Hello?
Thank,
Yeah,
thank you,
sorry.
Dr. Raj.
Okay.
It is a nice presentation, and good to know that arachidonic acid is playing a cytoprotective role against the alloxan and streptozotocin, and it maintains the blood glucose level, and it not alter the inflammatory mediators. It's a very nice presentation. Open for the questions.
In the chat box, there are some questions.
No. No, not yet.
Okay, all right. There is a comment. In the experimental animals where we performed both type 1 and type 2 diabetes elements, the animals which develop type 1 and type 2, there is substantial loss in the body weight. But when they are treated with arachidonic acid, the weight loss becomes normal. That means there is no further, that means they are almost normal weight, suggesting that because the diabetes is controlled, they regain their normal weight. But arachidonic acid control animals do not lose any weight, nor they increase in their weight. So arachidonic acid by itself, when given to the control animals, there is no change in their body weight. Is it question now? Okay.
I have one question. How about the DB mice? Because the streptozotocin is chemically induced model. So
Correct.
have you tried with the DB mice?
No, we did not try that anymore.
Okay.
We are of the opinion that genetic models are probably not very suitable for
Mm.
studying the diabetic pathobiology, because we are genetically inducing diabetes.
Yeah.
Whereas in humans, we are not genetically inducing diabetes. It is only because of the diet, lack of exercise, and so on and so forth.
Yeah,
So
good
that's
to know
the
you
reason.
saw the clinical data.
Yeah, and that is why we shifted to the clinic. So we started measuring lipoxin A4 levels, arachidonic acid levels in patients, and now we are planning to do a trial, see whether we can give arachidonic acid to these patients, or patients who are at high risk of diabetes, and see what happens to their plasma glucose levels and development of diabetes.
Oh,
But
nice.
the only problem we have is we have to give pure arachidonic acid.
Yeah.
Not like people give, for example, if you want to give study EPA DHA, you give fish oil,
So
which
you
are
have
rich
to,
in
you
EPA
have to
DHA.
give us a free fatty acid.
Yeah, we have to give a free fatty acid, and the 100% pure, because we have done an experiment with oils which are rich in arachidonic acid. We found that they do reduce the plasma glucose levels, but not as much as the pure arachidonic acid.
Yeah.
So now we are trying to work out the dosage that has to be administered to the patients, how frequently and how long, and so on and so forth. So a lot of pharmacokinetics and dynamic studies are to be performed, like classical pharmacology.
Yeah. Thank you.
Thank you very much.
Thank you, Dr. Ami, for the introduction. Today, I would like to give a talk on the lysophospholipid omega-3 fatty acids, which is a novel nutraceutical approach for the prevention of Alzheimer's disease. I would like to give a small background and a rationale about the Alzheimer's disease. Alzheimer's disease is the most common cause of dementia, which accounts 60 to 80% of the cases. The most symptoms of the Alzheimer's are difficulties with the memory and inability to learn new things. And they have the problem of solving things. And it also affects the person's ability to perform everyday activities. Alzheimer's disease is the sixth leading cause of death in the United States. But the one safe and cost-effective preventive approach is to increase the brain DHA levels through the diet. So, but however, the high DHA consumption is associated with the low incidence of Alzheimer's disease. But the prospective studies as using the current available dietary supplements have yielded the disappointing results thus far in both humans and animals. And I also would like to give a small introduction about the docosahexaenic acid because the DHA is a long chain omega-3 fatty acids and it plays a critical role in brain function, and its deficiency it is known to Alzheimer's disease. But the DHA deficiency, if it is provides a high dietary intake of DHA, it is negatively associated with the incidence of Alzheimer's disease. The brain is an important organ which is highly dependent upon the dietary DHA for its needs. There are various molecular forms of DHA are available. The most common molecular forms of dietary DHA are triacylglycerol, which is present in fish oil, whereas in phosphatidylcholine, which is present in krill oil. But still, however, the clinical trials of the current available preparations of DHA has failed to show in the AD patients and the animal models. So because of its inability to synthesize the adequate amounts of DHA from the precursors, because the brain has to acquire DHA through the blood-brain barrier. We hypothesize that this failure is due to the inability of the supplements to enrich brain DHA because since they are all observed in the form of DHA form. The DHA from the current available supplements is released as a free DHA, which is available as a fish oil or krill oil. It forms as a free DHA and finally it forms as a DHA, which does not cross the blood-brain barrier. But whereas, however, the DHA from the LPC, from the SN2 position or from SN1 position of PC, it observed as a phospholipid form and is transported as LPC, and it has the ability to cross the blood-brain barrier with the transporter of MFS T2A. So the overall objective of the study is to determine whether the enrichment of brain DHA by this novel nutraceutical strategy will delay or prevent the development of Alzheimer's disease in a mouse model of 5XFAD mice. So our aim is to test the hypothesis that dietary omega-3 is superior to the current available DHA supplements like fish oil or krill oil, which enriches the DHA in Alzheimer's disease mouse model of 5XFAD mice. So we had treated with the three groups. One we had used as a no DHA with the control like corn oil. The second group, which is a surrogate for the LPC, the TAG omega-3, and third group is like LPC omega-3. So the second aim is to suppose if it increases the DHA, so how it improves the brain function and whether it helps to prevent or ameliorates the AD pathology. Then we want to know the mechanisms. So we had studied the in vitro study also. So in this study, we tested the hypothesis that enriching brain DHA by LPC omega-3, which prevents or ameliorates the AD pathology using the 5XFAD mouse. This mouse model of Alzheimer's disease has been used extensively in the recent years to study the pathogenesis of Alzheimer's disease and to investigate the potential therapeutic interventions. It is a transgenic model which co-expresses the 5AD mutations, resulting in the overproduction of amyloid beta and accumulation of intraneuronal amyloid beta 42 by the age of 2 months. Since our primary goal is to evaluate the potential benefit of dietary omega-3 to prevent or delay the development of Alzheimer's disease, so this is look like resembling that of human disease in several aspects. That is the reason we have used this model for the Alzheimer's disease study. We next evaluated the effect of LPC omega-3 and the percentage of EPA and DHA fatty acids in the plasma and brain by GCMS. So here what we saw is that LPC omega-3 was effectively increasing the plasma omega-3 in 5XFAD mice. Then not only DHA, the EPA level also significantly increased the omega-3 in the plasma. And this, it replaces the arachidonic acid. So the similar trend has followed in liver. Here also the LPC omega-3 fed 5XFAD mice has significantly increases the omega-3 fatty acids in both EPA and DHA, and it predominantly predominantly affects the arachidonic acid. So in contrast to the liver and heart, the adipose tissue showed an increase in DHA concentration, and it also changes the omega-6 omega-3 ratio after the feeding of TAG omega-3, but not affected the LPC omega-3 fed 5XFAD mice. This result shows that the dietary TAG omega-3 is predominantly directed to the adipose tissue and heart in the form of TAG. But whereas the dietary LPC DHA may be directed to the brain in the form of phospholipid. And we have studied not only the fatty acid and of plasma and liver, we have studied in the brain also. So here our data of 5XFAD mice showed that the LPC omega-3 has significantly enriched the brain EPA and DHA levels. And it was significantly increased by the DHA levels was increased by 127%, and whereas the EPA was increased like 50-fold increase in 5XFAD mice fed LPC DHA as compared to TAG DHA. So to confirm that, we have studied the molecular species of LPC omega-3. So here the concentration of LPC omega-3 has increased significantly in mice treated with LPC omega-3 when compared to free DHA and control diet. So this, it shows that this correlates with the plasma fatty acid. So next we understood that like if there is an increase in the DHA, whether it improves the brain function or it helpful for the behavior. So we next determined if the higher plasma and brain levels of DHA after LPC treatment, whether it is associated with the improved behavior. So the behavioral studies was performed in collaboration with Dr. Leon. So here behavior analysis was conducted in the mouse dark cycle, tracked in a real time by an overhead camera, and videos analyzed by using the NMS software. So here we measured that total distance traveled and the time spent immobile were measured. So here results shown that there was no significant change from the control mice in any of the treatment groups. But we observed that the LPC mice spend less total time immobile than other groups. So this data suggests that the overall levels of anxiety-like behavior are higher in 5XFAD, DAG, and 5XFAD control mice. So I hope this is marginally modulated by the LPC omega-3 treatment. We next evaluated the effect of LPC treatment on memory relevant behavior using the novel object recognition test. Here, novel object recognition test, we have measured the time spent with each object and the discrimination index and preference index was calculated. In the 5XFAD mice, the performance in novel object recognition was followed by LPC omega-3. And here what we observed that the performance was 13% higher and 25% higher with LPC treatment when compared to 5XFAD mice of control and 5XFAD TAG mice. This data supports that LPC omega-3 treatment, it improves the memory relevant behavior in mice that co-expresses the human 5XFAD mice. So we want to confirm the one more behavior study, whether it is this DHA is beneficial for the brain function. So we have did that Morris water maze test, which is highly specific for the function of the hippocampus. And it is one of the most affected areas of the brain in Alzheimer's disease. In this, what we observed that the mice treated with LPC omega-3 showed significantly improved spatial learning and memory in the Morris water maze test. And moreover, that it has showed a remarkable improvement in the latency to the previous platform area, as well as the time spending in the target quadrant by the LPC omega-3, but not by the TAG omega-3. These results indicate that there was an increase in the brain DHA levels by LPC omega-3, which results in a marked improvement in the spatial learning and memory in 5XFAD mice. So next we measured the BDNF because BDNF plays an important role in the learning and memory, and it is a potential downstream target for the DHA. So therefore we determined the BDNF concentration in the brain regions by ELISA to complement our Morris water maze data. So here there was an improvement of brain function because of the increased levels of BDNF, which is known to play a critical role in learning and memory. We also found a significant increase in the BDNF levels after the LPC omega-3 treatment, but not after the TAG treatment. This shows a positive correlation of biochemical measurements with physiological changes. So not only the BDNF, we also measured the amyloid beta levels because the 5XFAD mice has a more overaccumulation of the amyloid beta. So we have measured the amyloid beta levels in the brain. So our results shown that the dietary LPC omega-3 has markedly decreased the amyloid beta 42 and 40 levels as compared to 5XFAD control and 5XFAD TAG. This indicates that the neuronal dysfunction occurs in the brain at the early stages and it progresses to encompass the brain with age. This also indicates that the brain has more sensitive to amyloid changes in the early disease. And the brain may be affected progressively with age. Next, we would like to study the mechanism because since inflammation is mediated through the microglia, it contributes the pathology in the Alzheimer's disease. So the dietary LPC DHA, which may inhibits the microglial activation and it may helpful for prevent or delay the development of disease in addition to promoting neurogenesis and neuronal repair. So here you can see this pathway. It has polarized to M1 and M2 microglia. So the M1 microglia, it produces with the LPS and IFN alpha, it polarized to M1 phenotype and produce the pro-inflammatory cytokines or mediators. It includes the IL-1 beta, IL-6, TNF alpha, CCL2, ROS and NO. Whereas the M2 phenotype, it produces acts as anti-inflammatory cytokine and it produces the resolvins and marasins and it acts as a neuroprotection. So Hence, the control of microglial activation and the suppression of pro-inflammatory mediators and cytokines in activated microglia, it provides the therapeutic benefits in the treatment of neurodegenerative disorders. So our results have shown that there was an increase to the BDNF expression in the microglia, and also it also inhibited the synthesis of the pro-inflammatory eicosanoids, and it also reduces the pro-inflammatory cytokines. So this indicates that LPC DHA is acts as an anti-inflammatory molecule. So I would like to summarize my part like the present studies demonstrate for the first time the feasibility of increasing the net concentration of omega-3 in the brain of Alzheimer mice brain through diet. And these results therefore have potential to provide cognitive benefits for the Alzheimer disease mice models due to increase in the brain DHA and BDNF. And more importantly, the mice treated with LPC omega-3 showed an significant improvement of spatial learning and memory in the Morris water maze test. And it increased the brain BDNF levels compared to DHA omega-3 treated 5-XFAD mice. So although these results need to be confirmed in humans, the present observations could lead to a novel nutraceutical approach for the prevention and treatment of neurological diseases such as Alzheimer's, Parkinson's, and other neurological disorders, which are known to be benefited by DHA enrichment of the brain. These are my collaborators which helpful for this study. Thank you. Thank you. Yeah, yeah, it is preventive. Yeah, I could not able to see the questions. Yeah. Yeah, since it is a dietary compound, so it is a preventive for some level. It's not like completely like drugs, it will prevent. We need to give more amount. So there is no commercially available of LPC omega-3. Yeah, it's a nice question. Actually, krill oil has phospholipid 60% of phospholipids, and it is present in the SN2 position. So it's again it converts as a TAG, triglyceride. If it is in SN1 position and if it converts to LPC DHA, that it may helps more for the brain function. Thank Yeah, thank you. The next speaker is Dr. Jankan Yogi. He is a professor at the University of Tokyo, Graduate School of Pharmaceutical Sciences. His field of interest, there are bioactive lipids, metabolic enzymes of lysophospholipids, receptors of for lysophospholipids, G protein signaling. He received his bachelor degree, Master of Science and PhD from the University of Tokyo, Faculty of Pharmaceutical Sciences. Please welcome Dr. Yogi.
Thank you for introduction. Can you hear me? And
yeah, able to hear and say,
okay.
Please put it in the presenter mode. Yeah, thank you.
Okay. So thank you for organizing for giving me an opportunity to present our data. We were focusing on lipidomics and lipid signaling, especially lysophospholipid signaling through G-protein coupled receptor. And I think this is this session is like a DHA session or fatty acid session. So I selected this unpublished study in the laboratory. So my first slide is here. And as many of you know, omega-3 fatty acid or N3 fatty acid, such as DHA and LPA, is very known for its role of heart protection. So the Inuit epidemiology study or Fat1 transgenic mice study suggests that omega fatty acid has a role in protecting heart from heart attack. So this is a reason why Japanese food is so famous and favorite by many people in all over the world. So as I said, omega-3 fatty acid somehow protect heart. Also, however, the molecular mechanisms are not fully understood. By the way, some nerve, such as vagus nerve or vagal nerve, this is a part of parasympathetic nerve, somehow protect heart. So this is a very famous study showing that lower vagal activity shows a poor prognosis. As you can see here, after a heart attack, patient with high vagal activity, the survival rate is well, but the patient with low vagal activity survival rate is very low. So both omega-3 fatty acid and vagal nerve somehow protect heart. So today I want to introduce you that the DHA lysophospholic acid, DHA-LPA, has explains these two roles. As you may know, lysophospholipid is a phospholipid with one fatty acid, and various types of lysophospholipids are present in the body. And over the past 3 decades, many G-protein coupled receptors have been identified for LPA, sphingosine phosphate SMP, and mainly from our laboratory, we identified GPCRs for lysopase, and recently GPR55 is reported to be a receptor for sugar-containing lysophospholipid, such as lysophosphoryl glucose or glycerol. So the structure of LPA is here. So LPA is very simple in structure. It has phosphate, glycerol, and fatty acids. And there are 6 types LPA receptors, named LPA1 and 6. And LPA is produced by major 2 pathways. One is autotaxin pathways from LPC, and the other is PAPILAN alpha or beta pathway from phosphoric acid. And very importantly, the second pathway mainly produces SN2, lysophosphotic acid, and also recent crystal structure of taxane revealed that this enzyme also like SN2 LPC containing PUFA at the SN2 position. So in my opinion, most LPA that activate LPA receptor is SN2 PUFA containing LPA. Through this pathway, LPA has many roles through its receptors and producing enzyme. And I don't explain all. There are many phenotypes have been identified through the study of knockout mice of these receptors producing enzyme and also using some inhibitors of autotaxin. The study of lysophospholipid are mainly focused on the identification of the receptor and the synthetic enzyme transporters, and functional study of knockout mice of their lysophospholipid related proteins, receptors, synthetic enzyme transporters. But much has been known from the study of using knockout mice, but we now want to know what's the role in human, that is, role of lysophospholipids in clinical level. So what's the strategy? So we are performing lipidomics analysis using two types of mass spectrometry. One is LCMS, which analyzes lysophospholipids in plasma or tissues. And the other is mass imaging, which can localize the phospholipids, including lysophospholipids in the tissue section. So the first LCMS is very simple, and using this method, we can measure the lysophospholipid in only 0.1 microliter sample. And the detection level is very high, and 10 picomolar level can be detected. We can, as you can see here, we can separate SN1 and SN2 lyso. Also, we can, of course, distinguish the various acyl chain containing lysophospholipid using this method. Also, mass imaging technique can visualize phospholipid species in tissue sections. Here, you can, as you can see here, some of the PE or PS species distribute quite differently in mouse embryos. So using this technique, we analyze various lysophospholipids, including diacylphospholipids, in human clinical samples, including plasma, urine, cerebrospinal fluid, or tumor, etc. And first, our choice to detect them is using mainly LCMS. And in collaboration with Doctors Yatomi and Kurano in the Hospital of University of Tokyo, until now, we have analyzed almost 10,000 clinical samples. And we found many changes in the level of lysophospholipid in various diseases. And precise data was recently summarized in this review. So please refer this. So today I want to focus on one disease, ACS, acute coronary syndrome, which is which contains a heart attack. And we, as you can see here, we found that some unsaturated LPA, such as 82 or 24 and 22.6 LPA. This is DHLPA is dramatically high in the plasma isolated from ACS patients. So importantly, most DHLPA detected was SN2 type with a little amount of SN1 DHLPA. So the maybe liphases or phospholysis N1 type enzyme are involved in this production. By the way, we also observed that in some neuronal disease, such as schizophrenia, SCG, or MDD, the DHLPA content in cerebrospinal fluid is significantly lower. So DHLPA may have some role in brain function as suggested by the previous presentator. So we want to know the role of DHLPA. So we first introduced mouse myocardiac infarction model, MI model. So in this model, we first ligate the coronary artery, and after 1 hour, we reperfused. And after 24 hours, we analyze phospholipid content by LCMS. So we distinguish non-ischemic area or ischemic area using some dyes. And we first examine the level of various phospholipids and lysophospholipids, and we found that DHLPA was dramatically increased in ischemic, only in ischemic area. Importantly, in the mouse model, plasma DHA level is not so high, and most of the DHA-LP was detected in the ischemic area of heart. So this suggests that DHA-LPA in a human clinical sample may derive from the heart after the heart attack. We also confirmed the presence of DHA-LPA using mass imaging. And as you can see here, unlike other LPA species, DHA-LPA presence is localized in the ischemic area, as shown in the left panel. So these two experiments confirm that DHA-LPA is produced in ischemic area after the heart attack in MI model. So our next question is, what's the biological significance of DHA-LPA? So interestingly, some of the lysophospholipid receptors appear to distinguish the SN1 or SN2 or PUFA or saturated fatty acid. And as you can see here, some receptors such as LPS3, LPS6, LPS1, or GPR55, these lysophilic GPCRs prefers PUFA or SN2 lysophospholipids. For example, this graph shows the ligand specificity of LPS3 receptors toward various types of lysophospholipid with different fatty acid. As you can see here, DHA-LPA, in this case, SN2 type DHA-LPA is the best ligand for LPS3 receptor, although some PUFA are containing or monounsaturated LPA is also a good ligand for LPA. So DHA-LPA is a potent ligand for LPS3 receptor. So in collaboration with Dr. Kanai and Shibasaki in the University of Tokyo, Japan, we synthesized a stable LPA analog named T13 because LPA has a free phosphate group which readily dephosphorylate in some enzymes such as lipid phosphate phosphatases. So LPA in general is very, very unstable. But in this case, sulf S is introduced in the head group, and this is a ring structure. T13 is dramatically stable, and it was found to be a very potent agonist for LPS3. So using this T13 as an LPS3 specific agonist, we try to analyze the role of 2,4-LPA in vivo. So to make the long story short, I explained the essence. So as I explained, upon heart injury or heart attack or in MI model, DHLPA is somehow produced, and this DHLPA activate LPS3 receptor in the vagal nerve, and it contribute to the heart protection in MI model. So we show some evidence. So first, activation of LPS3 receptor lowers the heart rate and decreases the blood pressure by our vagal nerve, and LPS3 mRNA is highly expressed in vagus nerve. Next we show these data. So in this slide, we just administrate T13, a potent LPS3 agonist I explained, in a mouse by IV injection. So just after the IV injection of T13, wild-type mice showed a very potent decrease in the blood pressure, and which accompanied the decrease in the heart rate. And these effects were never observed in LPS3 knockout mice. So this effect is on target, and LPS3 activation leads to the blood pressure decrease and heart rate decrease. So other than the hypotension and bradycardia, many pharmacological effect was observed, such as arrhythmia, low blood flow, and apnea, blood apnea, decrease in the O2 content, and a lower body temperature, like that. But all these pharmacological effect was induced when vagus nerve was hyperactivated. So we next examine if vagus nerve is involved in the T13 induced effect by inactivating vagus nerve, by cutting the vagus nerve or vagotomy. And if we don't cut the vagal nerve, M13 biological effect, pharmacological effect was observed as decreased by the blood pressure and heart rate, and which were never observed after the vagotomy. So this indicated that T13 effect was mediated by the activation of vagus nerve. We also, because vagus nerve is composed of two components, one is afferent nerve and it's efferent nerve, we use a specific reagent named capsaicin, which is a TRPV1 agonist, which specifically inactivates only the afferent nerve. So we administered capsaicin to the mice and tested the effect of T13. So as you can see here on the right panel, after the treatment of capsaicin, no pharmacological effect was induced by T13 in capsaicin-treated mice, suggesting that LPS3 agonist T13 activate the afferent nerve. In addition, the cell body of this neuron, afferent nerve, is present in some ganglion near the neck in the mouse and human, named nodose ganglion. So we performed in situ hybridization analysis of LPS3 receptors in nodose ganglion of mice. And as you can see here, some Berger cell body is very positive in LPS3 mRNA. So this clearly shows that this afferent nerve highly expressed LPS3 receptors. So we also examined the effect of ischemic damage on LPS3 knockout mice. And we found that in LPS3 knockout mice, ischemic damage became exacerbated. So as you can see here, the infected area after MI treatment in LPS3 knockout mice has more larger infected area compared with heterozygous control mice. And also, as you can see here, apoptotic cell, as judged by tunnel staining, the number of apoptotic cell increased in LPS3 knockout mice after MI treatment. Sorry. Also, when we treat the mice with LPS3, LPA1 antagonist KI compound, this is an LPA1, LPS3 antagonist. Similar effect was observed, as was observed in LPS3 knockout. Also, finally, we show that DHLPA or LPS3 agonist T13 ameliorated the ischemic damage. As you can see here, after the treatment, MI treatment, we also treat the mice with DHLPA or T13. And as you can see in this slide, treatment of DHA or T13 dramatically decrease the infected area or percentage of apoptotic cell in the tissue after the treatment. So this clearly indicated both DHLP and LPS3 agonist ameliorated the ischemic damage in MI model. So this is a negative control experiment showing that T13 effect was not seen in LPS3 knockout mice. So T13 effect was mediated by LPS3 receptor, possibly expressed on Berger nerve cell. So our data indicated that LPS3 agonist and endogenously DHLPA is a possible drug for protecting heart damage after a heart attack. So in summary, we showed that DHLPA is somehow produced after a heart attack or MI in the mouse model. So I want to emphasize that DHLPC is also elevated in this model. However, ATX inhibitor did not decrease the production of DHLPA in this model. So now we know that this path LPA, DHLPA is produced by ATX independent pathway. And I show that DHLPA targets the LPA3 receptor in the afferent vagal nerve cells, and it exerts its role in heart protection by activating efferent vagal nerve. By the way, we, in collaboration with Ono pharmaceutical company in Japan, we recently developed a supplement, lipid supplement named Lemware. This supplement mainly derived in salmon raw egg, and unlike other DHA supplement, this is very rich in DHA-PC, mainly 38.6 and 46. And in a mouse model or in a post-dosing in human, we found that DHA-PC level, the plasma DHA level is dramatically increased. This suggests that DHA-PC or itself, or after it is converted to DHA-LPC, they are directly incorporated into the blood through intestine and increase DHA-LPC level. So this supplement is now available for Ono Healthcare in Japan, and please try. So most of the work today we introduce is done by Unique Keno in the laboratory. And we also acknowledge various people here, especially Doctors Yatomi and Dr. Kanai in the University of Tokyo. Thank you for listening.
Thank you, Dr. Yogi. It's a very informative talk, and good to know that DHA-LPA is a potent ligand for LPA3, and it also protects the heart from ischemic damage. And good to know it is commercially available. Open for the questions. I have
Sorry, is this a question or?
I have one question. Like this LPA is helpful for the brain stroke, or how the?
Yeah, of course, yes. So I don't show the precise data today.
It is
this supplement changes the sleeping pattern. So REM sleeping pattern is quite different after we taking this supplement. So this is the name of this REM well. So sleep well or REM sleeping pattern change. This is the name of REM. So maybe I think the DHLPC in the plasma is increased after taking this supplement, and maybe DHA incorporation in the brain increased.
Okay, thank you. Thank you. We are moving on to the next speaker.
Thank you very much.
Thank you. Our next speaker is Dr. Lang Nam Gian. He is an assistant professor in the Department of Biochemistry within the Yang Lu Lin School of Medicine at the National University of Singapore. He received his PhD from the University of Hamburg in Germany. His lab in the National University of Singapore is interested in identifying and characterizing the molecules and physiological functions of the transporters for the lysophospholipids, such as S1P. With that, I will turn it over to Dr. Nguyen.
Hi. Can you hear me? Oh, okay, that's great. Yes. Yeah. Thank you so much for the invitation, Sugasini and the organizers. So yeah, my name is Long Nguyen from the National University of Singapore. So my talk today, I will give you some information about lysolipid transporters, but I will focus on like phenolgene 1-phosphate transporters that we have been working in the lab for several years now. So S1P, so as you can see here, this is a molecule um, in the lower part of the slide here. So this is the phenolgene 1-phosphate, and how it is actually synthesized in the cells. So this is actually the pathway. So we have like the de novo synthesis of the phenolgene backbone from the fatty acids, unbalanced acid, together with serines through a series of enzymatic reactions. So it will create ceramides. Okay, ceramide will be utilized for the synthesis of phenolgenes, and phenolgenes can be phosphorylated by two different kinases in the cells, and eventually it can make S1P, phenolgene 1-phosphate. In the cell, so we have also the the remodeling pathway. So from phenolmyelin, the cell can hydrolyze using the phenolene maize to make ceramide, and again, ceramide can be used to make phenolgene 1-phosphate. So remember that, so this is actually very common pathway for most of the cells, or all of the cells utilize the pathway to make phingolipids. However, so just a few cells that can release our S1P for signaling. So I will go through with you some of the cell types that we identify the transporters. For the function of S1P, basically it is an extracellular lipid mediators. So it releases out into the medium cells, and then it is an agonist for 5 different S1P receptors. So shown in here are 3 different receptors. So receptor 1, 2, 3. So they are G-protein couple receptors. So basically, how do they work? Is that they couple with like intercellular proteins called G proteins. So in this case, for the receptor 1, for example, it's coupled with GIs. And then what they do is that they actually induce the downstream signaling, for example, increasing like calcium level, increasing phosphokinase C, increasing like, MAP kinase phosphorylation proteins, and eventually what they do is actually they regulate, the cell function, like cell migration, cell proliferations, and so on and so forth. So basically, the function of S1P converts into S1P receptors, and they mediate the functions for lymphocyte trafficking, so that I highlight in here, or the blood vessel integrities, because so these receptors are actually expressed throughout the body, and they are highly expressed in the lymphocytes as well as in the cell in the blood vessels. So S1P signaling pathways have been identified for a couple of decades now, and there are a couple of drugs that have been developed for the treatment of disease. So in this case, the inflammatory diseases. So essentially, so these drugs like fingolimod or zalimos or the others targets S1P receptor in the lymphocytes. So basically, lymphocytes resides in the lymphoid organs. For example, in this case, it's the lymph nodes or in the thymus. So they have to migrate out, and in order to really exert the function, like clear the pathogens and so on and so forth. In order to migrate out, so in this case, for example, the lymph node, so they have to be triggered by S1P. So S1P release out in the lymphatic vessel in here, and then the cell, the lymphocyte can migrate out. So these drugs, basically, they target and they downregulate the expression of S1P receptor in the lymphocytes so that the lymphocyte cannot migrate out of the lymphoid organs. And by doing so, so they cannot traffic to the target organs, for example, in the brains. So that this drug can prevent like inflammation in the brains. So in this case, like multiple sclerosis or in the gut. So basically, these diseases are induced by reactive immune cells. And in this case, this drug can target the trafficking of immune cells to the inflamed site. So the problem for these drugs is actually they create a side effect, quite severe side effect, like slow heart rate and pulmonary leakage and even ocular edemas. So what happened to the patient is that so the patients usually they allowed to take the drug, but they have to stay in the hospital for a day so that they can be monitored for the heart rate. So if they pass through this quality check, health check, and they can actually take the drug. So if their heart rate have some problem, they have to stop from using the drugs. So this actually just severe side effect, and mainly it target the S1P receptor in the blood vessels. So I show in here some of the evidence why S1P signaling is quite critical for the blood vessel and why S1P receptor antagonists are actually showing the side effects. So you can see here two examples. So the first example is when the researchers indicate, the researcher from Timla group and Richard Pryor. So they knock out S1P receptor once in the mice, and you can see the knockout mice in here compared with the normal white type mice in here. So they have the leakage throughout the body in here. So the embryos are actually exposed in here. You can see the embryos have severe hemorrhage throughout the bodies. So the second example is a knockout of double kinases. So basically, we have two different fingosine kinases to make S1P, fingosine kinase 1 and 2. In order to see the phenotype, we have to make the double knockout for both kinases. So the double knockout for both kinases also have the severe phenotype, severe hemorrhage, which recapitulates the phenotype from S1P receptor knockout mice. So basically, if we abrogate S1P signaling, we will see severe hemorrhage because S1P receptors are expressed in the blood vessels, and they are very critical for the maintenance of the blood vessel health. So my lab is interested in lysolipid transporters. So we have been working with different lyso transporters over the past few years. However, I will focus my talk on the function of MFSD2B and SPNet2. So we have a couple of papers in here if you are interested in, so you can have a look. So for MFSD2B, so which is a homolog from MFSD2A, which I have been working on as well. And MFSD2A, as you know, it's LPC transporters, and MFSD2B is actually the exporter for S1P, mainly from erythrocytes and platelets. For SPN2, so far we think that it is expressed more in the blood vessels, like in this case in the endogenous cells, which release our H1P through this transporter, and both of them we think that contribute to the extracellular H1P for signaling. So for characterization of MFSD2B functions, so we generate the global knockout mice for these proteins, and first what we do is that we quantify plasma H1P levels, and as you can show in here, the white type mice, which usually have about 1 micromolar of H1P in the plasma, and when we knock out MFSD2B, the level of H1P reduced around a half. And most of the H1P that we can detect by mass spectrometry that's shown here are concurrently reduced due to MFSD2B knockout mice. So basically in the extracellular milieux, H1P are bound to HDL, so about 60% of H1P are bound to HDL, and about 30% are present in album, and both of the fraction are reduced in the knockout mice. So in order to see which cell types which are expressed with the proteins MFSD2B, so we knock it out. We knock out MFSD2B in erythrocytes. So in here, you can see that when we use the conditional knockout mice to generate the knockout mice, and the protein band is absent in here compared with the wild type mice. So in this case, in erythrocytes. So we then generate also the platelets knockout mice, and as you can see, the protein is also absent in the platelets. So we wanted to see which cell type are the major cell types that contribute to H1P levels, and as I summarize in here, so this is the wild type mice, the global knockout mice have about 50% of H1P reductions, and as you can see, when we knock out MFSD2B in erythrocytes, and H1P level reduced about a half. So in this case, so we think that H1P released out from the erythrocytes is probably the major source contribute to plasma H1P. So next, we wanted to see whether MFSD2B which release our H1P, whether it's important for regulation of lymphocyte trafficking. So we could detect a slightly reduction of lymphocytes like CD4 positive or CD8 positive cells. They are T cell here, or B cells. So basically, H1B level from MFSD2B is not very critical for regulation of lymphocyte trafficking. So we wanted to also check whether the source of S1P from MFSD2B is also important for regulation of blood vessel integrity. So in this case, we did a very simple experiment where we inject with even blues, it's a dye into the circulation system, and we wanted to check whether the dye extravasates into the tissue, but we couldn't really see much of differences. So in this case, we conclude that even though with the reduction of S1P, so the blood vessel, the circulation system of the knockout mice still quite intact. So as I mentioned quickly, so there's also another S1P transporters that have been identified by several groups, like the group from Dr. Makyosuke in Japan. So essentially, they identify this transporter from zebrafish, and when they knock it out in the mice, so they measure S1P level in the plasma, and they could show that S1P level in SPN2 knockout mice also reduce a half. So however, this level of S1P contributed by SPN2, it's a little bit contradict in several studies. S1P level in the plasma from the knockout mice was slightly drops. In some study, even couldn't detect the reduction of S1P due to SPNA2 knockout mice. So we wanted to see whether MFAD2B and SPNA2 are the intracellular S1P. So in this case, so we generated the double knockout mice in order to eliminate the hypothesis that there are also additional S1P transporters. So when we generated the double knockout mice, so in this case, the two genes are completely removed from the mice. And as you can see, the mice are here at the very early stage, at the embryonic stage. So they have hemorrhage. And as you can see, clearer at the later, 1 day later in the embryos in here. So as you remember, so I show you that knockout S1P receptor 1 or S1P synthesis enzymes. So both of the models have also severe hemorrhage, and we could recapitulate the phenotype by knocking out both transporters in this case. So we wanted to check also the blood vessels, and we was interested in looking at aorta. Aorta is the main artery in the body. It's the biggest vessel in the body. And you can see it's a very nice structure of aorta in the wild type animal, in the control animals. And the aorta of the knockout animals are collapsed. So basically, they cannot maintain the function of aorta due to the low level of S1P. And we found somehow the high, increased proliferations of endogenous cells shown in green in here, shown by CD31 staining. And the integrity of the aorta actually is weakened. It's ameliorated, as you can see that when we stain with Claudin-5, which is a tight junction between the cells, and the expression pattern of Claudin-5 is actually dysregulated in the case. So the data shown here suggests that S1P from the two transporters are important for maintenance of the blood vessels. So next, we wanted to see whether both transporters also contribute to S1P in adult stage. So in this case, we generated the double knockout at the adult stage. So we have a way to do so. We use MH1 to knock out both transporters. And in order to induce MH1, we have to inject with a drug called poly-IC. And after a while, we collect S1P for analysis. So in order to confirm the good deletion by this strategy, so we show in here, MFA2B is actually absent from the double knockout compared with a single knockout. For SPNA2 knockout, because it is not expressed in red blood cells, so basically we can still see the expression of MFA2B. And in order to see the knockout of SPNA2, we use a surrogate marker. We use lymphocyte level, because when we knock out SPNA2 in the mice, in the single knockout, this is single knockout, so we could see the reduction of lymphocyte. This is wild type animals, and this is a single knockout for SPNA2 in comparison with the double knockout, whereas single knockout for MFAD2B is slightly reduced lymphocyte level in blood. So in this case, we think that we have a good knockout system. Next, we wanted to see the plasma S1P level in the double knockout, as I show you in here. So you can see the wild type animals and the single knockout for SPNA2. In this case, we don't see a reduction. For the single knockout of MFAD2B, we could see about 50% of reduction of S1P. And when we measure S1P level in a double knockout, it reduced about 80%. So what are actually the significance of these pathways, of these two transporters for extracellular S1P for signaling? So in this case, we wanted to test for the blood vessels. So we use histamine to induce some kind of like allergies, anaphylaxis. So when we inject a dose of histamine to the wild type animals, so the animal core body temperature drop, but they could recover nicely, whereas the body temperature of the double knockout decreased, but they could not recover. So more than like 75% of the knockout mice die because of a dose of histamines. So this data suggests that H1P is important for maintenance of core body temperatures. So when we injected with a dose of H1P, so basically we conjugate H1P in BSA and we injected to the circulation system of the animal, and it could recover the body temperature from the double knockout shown here. So we also wanted to check the phenotype in the lung because lung vasculature is very important. And as you can show in here, is the lung histology from the control animal, and this is from the double knockout mice. So as you can see, some of the cluster or clots of the blood inside the vasculature inside the lung in here, and which show that the lungs are actually quite leaky. So they have like, increased level of even blue leakage into the lung. So basically, when we remove MFAD2 and SPNA2, we reduce H1P level, and the reduction of H1P level in the animals affect the vasculatures of the animals. So, so far I show you that the two transporters are the only ones that we think provide H1P for signaling. And we were very interested in the fact that SPNS2, even though it doesn't contribute much of plasma H1P, but they have a very significant role in reduction of lymphocyte level in blood. So as I mentioned that if we disrupt H1P signaling, we basically block lymphocyte egress from the lymph node, right? So in this case, we remove this protein and it could also recapitulate the phenotype. So we generated also the global knockout and also the conditional knockout for SPNA2 using MS1-CRE. I forgot to mention that MS1-CRE is only target blood vessel cells, hepatocytes, and also hematopoietic cells. However, SPNA2 is only expressed in the blood vessel cells, so it doesn't really express in the hepatocyte or hematopoietic cell. So in this case, it targets quite specifically in the blood vessel. And as you can see in here, so the lymphocyte level actually reduced to the level that we could achieve with the global knockout. So S1P, SPNA2 completely removed from the mind in this case. So first, we wanted to measure how much S1P is actually released out from the limbs. So let me explain to you how it works for this experiment. Okay, so this is just a piece of lymph nodes. So basically, S1P actually released out in the lymph here from the lymphatic vessel, from the endogenous cell here. And in this case, we collect the lymph inside here, so very tiny amount, so that we can measure H1P level in here. So we collected the lymph from Chitana Kali. So as you can see, it looks quite milky because they don't have blood cells. And so the lymphatic vessel is important for trafficking or transport of lipids, as everyone knows, right? So when we measure S1P levels, so as you can see, S1P level in a wild type animal in the lymph, so it's about 0.2 micromolar, 200 nanomolar, which is about like 1/4 of what we found in blood. However, when we knock it out, SPNA2 S1P level in the lymph is actually dramatically reduced. And what happened to it? So we check the lymphocytes in the circulatory system. So we counted white blood cell. So there are no reduction for other cell type like red blood cell and platelet. So when we check for CD, CD3, CD4 T cell and CD8 T cell, so the animals also have significantly reduction of S1P lymphocyte level, as shown in here for T cell and shown here for B cells. So it's quite interesting to note that for the lymph node structures, so basically when we remove SPNA2 completely from the animals, so it actually affects the lymph node structure. When we temporarily remove SPNA2 in the conditional knockout, the lymph node size in this case is rather conserved. So what is actually the significance of this pathway? So in this case, we apply some disease condition to the animals, right? So to the wild type animal and to the knockout animals in order to see the significance of the proteins, the genes. So in this case, we apply neuroinflammation. So we injected with an antigen that can induce neuroinflammations. And in the wild type animals, so basically in this model, they have paralysis. So this is actually the model for multiple sclerosis. And the disease score shown here is higher, so they have more disease. So the wild type animal have more disease. And as you can see, when we knock out the transporters in the whole body, whole body, or in the blood vessels only, the disease score is actually dramatically decreased. So basically, they are protected from the disease. So when we measure the pathogenic T cells, so in this case it's T17, Th17. So this pathogenic T cells actually decrease in the brain. So basically they cannot infiltrate in the brain to induce neuroinflammation like in the wild type mice. And we could see, a clearly phenotype, the protective phenotype in the two knockout mice in here. For the myelination, we did a very simple experiment to stain with luxol fast staining, and we could see that the myelination of the animal are still quite preserved. So this is the first model, and then the second model that we recently developed. So we was interested in the gut inflammation as well. So in this case, we induce gut inflammation by treatment with a dose of DSS. So as you can see, this is non-treatment for the wild type and conditional knockout mice. And if we give a dose of DSS in the wild type mice, so the colon is actually shortened and they have severe bleeding and also phenotype. And this is somehow protected in the conditional knockout mice. So as you can see some of the phenotype for the wild type mice, you can see in the colon here, when they have severe bleeding, usually the villi are actually fused, and you can clearly show in here. And for the conditional knockout mice, so the colon histology are actually quite preserved. It's not the level as a wild type mice, but somehow it's protected. And we could see like a reduction of inflammation. Basically, in this case, we use STAT3, which is actually a transcription factor, which one is phosphorylated, it can induce inflammation. You can see an increase of these proteins in wild type mice under DS treatment, and which is actually reduced in the conditional knockout mice here. So we also look at others marker for inflammations, and you can see here NF-kappa B expression also reduced in the mice, in the colon of the conditional knockout mice. So basically, we think that knockout SPNA2 can prevent lymphocyte infiltration. So we have more data. So we have almost a manuscript now. We think that it prevents lymphocyte infiltration to the inflamed site so that they can induce inflammations. So that's it. So let me wrap up. So basically, we show that MFAD2B contributes about 50% of H1P from red blood cell and platelet, and SPNA2 contributes about 20 to 25% from endogenous cell, and we think that both transporters are the only one that provide H1B for development and in adult state, so they contribute about 80% of H1B for signaling. And we hypothesize that during the adult stage, there might be also additional H1B transporters that have not been identified. And we think that targeting SPN2 could be used as a drug target for the treatment of inflammatory diseases. And we also published a paper that we show the vasculatures and cardiovascular function of these knockout mice are quite preserved. So we think that targeting SPN2 are actually better than targeting the H1B receptor to preserve the cardiovascular functions. So that's it. So I would like to thank a lot of former members shown in here. So like Thiet Vu, who identifies the function of FFNE2B, Toan, and is the current member in the lab. And I would like to thank also the funders, NMRC, MOE, and NOS and NOH, which support for my studies. So thank you so much for listening, and I will be happy to answer any questions that you might have.
Thank you, Dr. Nagian, for the informative talk. And good to know that S1P from SPNS2 is playing a major role, and it is treatment for the inflammatory diseases without compromising the vasculature. Open for the questions. So you can see the question, how LPA level is increased during the heart attack. 1%.
No, this is not my question, so I think. Look like they have no questions. Right.
Yeah, thank you, Dr. Nagian. Yeah. Thank you, Dr. Nagian. We are moving to the last speaker. Our last speaker today is Dr. Robert Sargis. He received his BA in chemistry from Carleton College in Northfield, Minnesota, prior to completing an MD PhD at Rush University, where he studied lipid and lipoprotein biochemistry under the mentorship of Dr. Subbiah. Rob completed his internship and residency in internal medicine, as well as a fellowship in endocrinology, diabetes, and metabolism at the University of Chicago, where he studied the adipose physiology under the Matthew Brady. Rob was on the faculty at the University of Chicago before being recruited to the University of Illinois at Chicago, where he is now a tenured associate professor in the Division of Endocrinology, Diabetes and Metabolism, as well as the section chief of endocrinology at the Jesse Brown Veteran Affairs Medical Center. In addition to his research, he is examining the impact of endocrine disrupting chemicals on metabolic physiology. Rob has an active clinical practice focused upon caring for patients with diabetes, lipid disorders, and other metabolic diseases. So I welcome Dr. Robert.
Thank you, Dr. Dhavamani. Can you just want to confirm you can hear me and that the slides look good?
Yeah, able to hear. It's good.
Excellent. So I'm super excited to present this work, which is really just beginning. So all of the data that I'll present today is basically preliminary work that's moving hopefully toward publication sometime soon. By way of disclosures, I do some consulting work. None of it impacts today's discussion. But what I am going to talk about is a project that fits well within the mission of my lab, which is to understand how humans alter the environment and how those alterations impact human health. And as a practicing clinician, our ultimate goal is really to translate this knowledge into action to protect metabolic health and then the consequences of metabolic disruption on other aspects of physiology. So this evening's scope, what we're talking about sort of broadly are metabolism disrupting chemicals. So these are basically exogenous chemicals or mixtures of chemicals that interfere with any aspect of metabolism. So these are conceptually a subset of endocrine disrupting chemicals. Which have been linked to various disruptions in hormone signaling, including alterations in estrogen and androgen and thyroid signaling, and have increasingly been implicated in the pathogenesis of metabolic disorders such as obesity and diabetes. Critically, given the centrality of energy metabolism to diverse cellular functions, that means that these metabolism disrupting chemicals are also implicated in other aspects of toxicity. So this includes diseases such as cancer and neuropsychiatric disorders, which is sort of where we're going to end up by the time I'm done tonight. Our lab has principally been focused on understanding how environmental toxicants contribute to the development of diabetes. And just so that we're all on the same page, I think it's important to recognize that when we talk about diabetes, what we're talking about is a disease that's defined phenotypically by elevations in blood glucose levels. As a consequence, this is really a constellation of diseases with various pathogenic origins. But the basis of this is really defined by the slide on the right, in which most people's glucose homeostasis is maintained in a normal range through the matching of insulin sensitivity shown on the x-axis and insulin secretion on the y-axis. So, if we find religion, we start exercising, we take care of ourselves, we sleep, we eat better, we become progressively more insulin sensitive, and our body makes less insulin to maintain blood glucose levels in the tight glycemic range. The problem is that most of society, and I think the data that's come out even this week is that 50% of the world will be overweight or obese in the coming years. Many of us, as a consequence of that, gain weight, are less physically active, and sleep less, and become progressively more insulin resistant. In the acute setting, that's fine. Our beta cells compensate by making more insulin to maintain blood glucose levels in that tight range. But that stress on the beta cells ultimately leads to their failure, ultimately resulting in impairments in insulin secretion during periods of peak demand, and when one develops impaired glucose tolerance, and ultimately even during periods of fasting when one develops type 2 diabetes. So the important thing is that when we conceptualize how one develops type 2 diabetes, or even type 1 diabetes to some extent, it really is this mismatch of insulin resistance coupled with beta cell failure, with beta cell failure being central to the development of hyperglycemia. Now, what's important about diabetes is that this is a disease on the march. So we're seeing more disease, we're seeing it in younger people, and it's important to also recognize that when we talk about diabetes, we're talking about disease that's not just a disruption in glucose homeostasis, but also disruptions in other parameters as well, including lipid disorders. So what's important to recognize, and this is sort of really my disclosure slide, is that various things contribute to the pathogenesis of diabetes. So clearly underlying genetics lead to increased susceptibility, as does physical inactivity and poor diets. The fact of the matter, though, is that these factors alone fail to fully account for the magnitude and rapidity with which diabetes rates have increased across the globe. So my lab has been principally interested in understanding how other factors contribute to metabolic deterioration, specifically how environmental exposures may be contributing. So On the left here is a simple correlation that we put together in 2011 that I think summarizes a couple of key points that I want to emphasize. So what you see here in red are diabetes rates in the United States from the late 1950s until 2008. And what you can appreciate from this is the dramatic increase in diabetes rates over this period. These increased rates reflect global changes in diabetes as we're seeing increased prevalence of the disease across the globe. But what's superimposed on this graph in blue is synthetic chemical production in the United States from 1938 to 2008. And what's remarkable about this is sort of the concordance between these two curves, in which we're seeing really dramatic changes in the types, materials, and environmental release of these chemicals that humans are exposed to. These are unique chemicals to which people have not been exposed to across human history. These chemicals fall into a variety of different categories. So these include chemicals used in food production that are generated through industrial activity. These are chemicals that we choose to put in our bodies and use in our homes, including personal and home care products. And this is sort of a subset that's of particular interest to me. There are synthetic chemicals that we use in the course of medical care that have also been shown to induce insulin resistance and impaired beta cell function and be linked with diabetes. So what's important here is that the totality of the data really suggests that an increasing number of environmental contaminants are linked to disruptions in glucose homeostasis. So this includes chemicals linked to insulin secretion as well as insulin action. I was excited to see Dr. Das's talk talking about alloxin, which is the first synthetic chemical shown to disrupt glucose homeostasis in 1943. But over the course of the last several decades, a number of different chemicals have been identified to disrupt glucose homeostasis and metabolic physiology. And these include both inorganic and organic compounds of both natural and synthetic origin. We've studied two, talafluanid and arsenic, and it's the latter that I'm going to talk about. But what's important when we look at the totality of the data is that really the diversity and consistency of that data suggests that toxicant exposures represent novel diabetes risk factors. And importantly, disparate exposures to these chemicals are also likely contributors to health disparities as well. But while our focus has generally been on diabetes, I think it's important to recognize that lipid disruptions are also a possible consequence of not only disruptions in this relationship between insulin secretion and insulin action, but also other metabolic disturbances as well. And this is an area that needs more study. Importantly, while at some point it gets a little depressing to think about the fact that each of us has 2,000 to 3,000 unique chemicals in our blood that may be linked to metabolic dysfunction, it's important to also recognize that this actually represents an opportunity. If we can change our practices to reduce exposures, we have the capacity to reduce risk in ways that we currently are not leveraging. So I want to focus today on a chemical that we've been studying now for several years, and that's arsenic, which is really a tremendous threat to global health. Arsenic is a metalloid that exists in various forms in nature, including different oxidation states, inorganic forms, and organic species. And why it's important is that it contaminates the drinking water for somewhere between 90 and 220 million people across the globe. In addition to being a natural contaminant, it's also historically been used as a pesticide, with some arsenicals still used in poultry and pig industry. It's also found in seafood and various agricultural crops, the most important which is probably rice. And then other exposure sources include things like coal ash, hydraulic fracking, and forest fires, which links arsenic exposure very clearly to climate change, both its origins and its consequences. And then as someone who works at the VA, I'm particularly interested in military-associated exposures, of which the most significant for US military personnel, as well as those living in Vietnam, is that the US military sprayed somewhere around 4.3 million liters of arsenic containing pesticides by hand during the Vietnam War. And the data linking arsenic to metabolic disruptions and cardiovascular disease is relatively robust. So it's linked to these disorders by cell-based assays, animal studies, as well as population-based studies. In our own work, we have shown that when we expose adult male C57 black 6 mice to arsenic and achieve levels in the liver that are comparable to highly exposed populations in Bangladesh, what we see is that these mice develop glucose intolerance, which is shown here in this intraperitoneal glucose tolerance test, in which the arsenic exposed mice get higher blood sugars that stay higher longer. And in follow-on studies, what we've shown is that this results as a consequence of decreased insulin secretion that likely occurs for a variety of reasons. When we started to get into this space a little bit, there were a couple of other things that sort of jumped out to us. And one of the interesting things is when we looked at these mice during metabolic cage analyses, what we saw was a shift in the circadian rhythm of food intake, favoring even more food consumption during the light cycle relative to the dark cycle relative to the light cycle, despite the fact that total food intake was not changed. So this raised some interesting thoughts about whether this arsenic was actually working centrally, in addition to its peripheral effects on insulin secretion. And then in the course of these studies, we did a couple of things that we standardly do, which is basically look at the effects of arsenic in the context of secondary metabolic stressors. So in this experiment, we exposed adult male C57 black 6 mice to arsenic at the same concentration in their drinking water in the context of a high-fat diet in which the fat was 60%. And lard in origin. And what was interesting in these mice is that we saw an actual decrease in body fat. And this was seen both by DEXA scanning as well as at necropsy when we actually weighed out the different adipose depots. And I think what was most striking is when we look then at the arsenic levels in the liver and how they correlated with adiposity, we saw a marked concordance between these curves. There was a tight relationship between liver arsenic levels and visceral adiposity. And this is total visceral adiposity, and this is specifically the mesenteric depot. And this decrease in lipids got us thinking about what this could possibly mean, not just in terms of total lipids, but what these lipid species may be. And through collaborations with Dr. Dhavamani and Dr. Subaya, we've sort of gone now down this road trying to ask the question of how this really important environmental toxicant may be influencing essential fatty acids. Now, you've heard about this all tonight, so I don't really need to belabor this too much, but it's important just to recognize that essential omega-3 fatty acids are critical for brain function. So DHA plays a critical role as an anti-inflammatory, antioxidant, and neuroprotective agent. It's important to recognize that it's enriched in neurons, despite the fact that they can't synthesize it, and that in population-based studies, those who have higher levels of omega-3 fatty acid consumption have a lower risk of depression. Conversely, in rodent studies, omega-3 fatty acid deficiency increases depressive-like symptoms in rats. And we know that other stressors that are associated with depressive behaviors also decrease brain DHA levels, including interventions like developmental restraint stress. And so thinking about this, it's interesting to contextualize then the established data looking at arsenic, in which there's evidence that arsenic and mental health disorders may be linked. So in animal models, arsenic accumulates in the brain and induces depression-like behavior. Developmental arsenic exposure has been associated or shown to induce cognitive impairments and depression-like symptoms in rodents. And it impairs hippocampal neurogenesis, induces disruptions of the hypothalamic pituitary adrenal axis, and alters brain-derived neurotrophic factor production. And then when we look epidemiologically at human populations, higher arsenic exposure is epidemiologically linked to multiple neurocognitive disorders. So this includes behavioral disorders, mood disorders, as well as cognitive disorders. So based on sort of this collection of data, we were interested in understanding how brain levels of essential fatty acids might be altered in our mouse models. And so in collaborations with Dr. Sabaya and Dr. Dhavamani, we measured DHA levels in the brain. And so what we see here is I'm just sort of narrowing down to the essential fatty acid, the fatty acids of greatest interest for the purposes of this talk. And what we see is in these adult mice exposed to arsenic in their drinking water for 8 weeks at a relatively high concentration, but again, a concentration that leads to internal levels that replicate levels seen in highly exposed human populations, we see this dramatic decrease in total brain DHA levels. This reduction in brain DHA levels is also, we see concomitant with that, an increase in arachidonic acid levels as well. So this is a shift in sort of a shift toward more pro-inflammatory, we would say, fatty acids versus anti-inflammatory fatty acids in the brain. Now, we also did these studies using a developmental exposure model in which we exposed C57 black 6 mice to arsenic in their drinking water at a concentration significantly lower. So in this study, this concentration was 100 times less at 500 parts per billion. Their exposure was for 4 weeks was from 4 weeks prior to gestation through gestation day 14.5, at which point the mice were sacrificed and we measured fatty acids in the brains of the dams as well as in the brains of the fetuses. And what you can see here shown on the left is that in the dams, after this relatively short period of exposure at this much lower concentration of arsenic, we saw a marked reduction in brain DHA levels in the dams as well as in the pups. And then when we looked at arachidonic acid levels in the dams, we saw the increase in arachidonic acid levels, which wasn't recapitulated in the fetal heads. In contrast, however, those fetuses did show increased expression of TNF-alpha in the brain. So given the importance of these fatty acids in the brain and the potential links between arsenic and neurocognitive disorders, we were really interested in the work of Dr. Sabaya and Dr. Dhavamani that had shown that brain DHA levels could be enriched using lysophosphatidylcholine DHA. And so Dr. Dhavamani did an outstanding job showing you all her data, looking at the Alzheimer's models. But this is just basically an important summary is that in normal mice, supplementation with LPC DHA can enrich brain DHA levels, whereas free DHA does not, and that this enrichment in brain DHA levels is associated with an increase in BDNF levels. So our interest was then in understanding whether this treatment could potentially be leveraged to address environmentally derived disruptions in neurocognitive function mediated by arsenic exposure. So our working hypothesis, which I think is still working, is that arsenic depletes brain DHA levels, which leads to a variety of molecular disruptions that ultimately lead to neurocognitive and endocrine dysfunction, and that specific repletion with LPC DHA, but not TG-DHA, can enrich this brain DHA levels and restore neurocognitive function. So to do this, we used a developmental exposure model in which we exposed C57 Black 6 dams to arsenic from 2 weeks prior to gestation, throughout gestation and lactation, and then randomized them to either a vehicle diet or LPC or TG supplemented DHA at the same molar concentration of DHA. Primary outcome was looking at behavioral in the pups, where dams are considered the experimental unit to account for litter effects. And then we looked at the dams, behavior in the dams as an exploratory outcome without any dietary manipulation. And what we saw in these mice is that there was no effect of arsenic on litter size or sex ratio. There was a very slight increase in weaning weights in both males and female offspring. That was very tiny in terms of magnitude, and likely the significance is driven by the number of mice in the study. When we looked at the dams, one of the things that we saw that was interesting is that in a novel object recognition test, those arsenic exposed dams studied 2 weeks after the completion of arsenic exposure had reduced exploring time, both for familiar objects as well as for the novel object. And those same dams had an increase in freezing time in fear conditioning testing in the contextual phase of fear conditioning. This is a phenomenon that may recapitulate phenotypes of post-traumatic stress disorder. So we're really interested in understanding how this specific exposure during pregnancy affects long-term outcomes in the dams, knowing that pregnancy is a unique period of vulnerability for environmental exposures. In looking at the offspring in cued fear conditioning studies, we saw a reduction in percent freezing time with arsenic exposure that we believe reflects an impairment in learning, although we have more studies to do in this regard. That impairment in freezing time, that impairment in learning and memory was reversed by LPC-DHA, but not by TG-DHA. And then when we looked at contextual fear conditioning, we saw a similar reduction in what we're calling a learning and memory deficit with the arsenic exposure that was partially reversed by the TG-DHA, but fully reversed and even above the controls with the LPC-DHA, suggesting that this LPC-DHA may be a unique means of addressing toxicant-induced disruptions in neurocognitive function mediated by these alterations in essential fatty acids in the brain. This is important because in many instances, arsenic exposure cannot be avoided. And so, in environmental toxicology, our goals are always to reduce exposure, but certainly we need efforts to address some of the permanent deficits induced by prior exposure, as well as means of addressing the needs of those who can't escape exposure. Current work in the lab is really focusing on understanding both the neurobehavioral outcomes that I've sort of alluded to already with the previous data, as well as looking at the mechanisms that may be responsible. And our hypotheses revolve around several things, including oxidative stress and inflammation, alterations in the HPA axis, which is particularly interesting from a veterans health standpoint as we think about disruptions in the HPA axis being a component of post-traumatic stress disorder for exposed veterans, alterations in neurotrophin secretion, and then also changes in serotonin biology, where our previous studies have shown that arsenic has the capacity to disrupt serotonin levels in pancreatic beta cells. We think similar effects may be occurring in the brain as well. And what's important is that anytime we study these toxicants, the critical point here is that likely multiple pathways are involved. Toxicants aren't drugs. They likely affect multiple pathways. The questions are what are the most relevant and important pathways, and how may we address them in addition to the interventions that we've discussed. So the shift in the lab has, thanks to the help of Dr. Dhavamani and Dr. Subbiah, has really been to understand how these environmental toxicants disrupt metabolism, leading to neurocognitive dysfunction. And as I alluded to before, as a clinician, I'm particularly interested in how some of these metabolism-based interventions may be used to address the environmental toxicity so that we may be able to improve human health. And importantly, these environmental toxicants fit within a broader realm of factors that disrupt developmental programming. So thinking about how these exposures may synergize with secondary stressors to induce neurocognitive disruptions, and how much of that may be mediated by alterations in brain lipid metabolism, and how that may be addressable using nutraceutical approaches. So with that, I'm going to close. Just thank the group, thank our funding, thank you all for tonight. I really appreciate the opportunity. This is a new area for us, for sure. I'm super excited about it, and would certainly be welcome to any feedback, suggestions, and those types of things. And with that, I'll close by just a quote from Maya Angelou, which is, "I've learned a lot, and most of it is I still have a lot to learn." So I appreciate all the wisdom that you can impart on me. So thank you so much for your time and the opportunity tonight.
Thank you, Dr. Sargis. Since arsenic exposure is linked to multiple neurocognitive disorders, I hope that LPCDHA is reversing by it and improve the cognitive function, and happy to continue the collaboration. Open for the questions.
Yeah, we're super excited about it. It's and our other thing is that, you know, this is probably not just unique to arsenic. So arsenic is probably the most important from a public health standpoint, from a global perspective, but it's likely the case that other pro-oxidant toxicants like may induce similar effects. So when we think about the potential clinical applicability, it's broader than this. I mean, we can think about other people who are exposed to other pro-oxidant toxicants during their occupations or environmentally, you know, that may have higher risk of even things like Alzheimer's disease, in which these types of interventions may be particularly important.
Yeah.
And then the other thing I would say while we're waiting for questions is, you know, the interesting part of this, and where the LPC-DHA is going to be, I think, particularly interesting, is that some of these toxicants accumulate in the brain. So one of the questions is, you know, A, can we reverse prior exposure, but can we prevent adverse effects? So use it as sort of a preventative agent as well as a salvage agent. And how would that potentially work if some of these metals accumulate in the brain and this oxidative stress is sort of an ongoing process? So these are, I think, some really interesting questions that are still lurking ahead of us. There's a lot of work to do, and it's hopefully exciting.
We need to look into it, more mechanisms.
100%.
We can wait for a few minutes for the questions.
Everybody's run out of steam, I think. It's been a great seminar, though. It's great. The talks have been wonderful. I'm really delighted to have been a part of it.
Thank you, Dr. Robert. I think no more questions, I think. I would like to wrap up the session. Thank you. Yeah, thank you so much. Thank you again for attending our event today. I hope you enjoyed it. The recording of today's webinar and the certificates for those who registered and attended live will be sent out later this week. Thank you again and have a good day.